Hello, everyone. It's noon sharp our time. I quickly started to welcome you all to this animal-free safety assessment collaboration workshop dedicated to the monocyte activation test. I'm Laura Viviani. I'm a consultant at SciEthic, working here on behalf of Humane Society International. It's a pleasure to see you all being here with us today for this very important workshop. I would like to just share a few slides and kicking off the meeting officially. So some initial rule for you online. So the participants will be in the listening mode only. Only the speakers and the panelists are able to unmute themselves and intervene. So please be active in the Q&A session of Teams. We will take your questions at the end of every presentation if there is time. Otherwise, we will try to answer it either online through the Q&A or through the chat, or we will bring up your questions during the panel discussion at in the last part of this workshop. In case we are not able to take all your questions, please apologize in advance, but we will try to be ready to all your questions while the event goes along. Please again make sure that to use the Q&A if it's active on your end. Otherwise, we will monitor also the chat as well. For your information, this event is going to be recorded, and the recordings and the presentation will be available on the AFSA event page. So we will make a post on LinkedIn and online to share when the presentation and the recordings will be available. So coming up to today's events. So we would like actually to give you a current overview about all the activities and projects that are ongoing at the global level and at the local level to implement the monocyte activation test as a replacement for the rabbit pyrogenicity one for the biological products. You will hear experts explaining what they have done to implement the MIT for blood-derived products and also for vaccines. We would like to offer you a platform where various stakeholders can really provide you a practical support and exchange with you what they have done and how important it is to have a product-specific validation in order to be successful with the implementation of this technology. We would like to make sure to hear your challenges, your issue, but also your experience if you want to share something. And hopefully we will give you an interactive experience. And hopefully we would like to give you an idea about what does it mean to phase out the rabbit pyrogenicity test from a global, regional, and local perspective. We are actually working globally on supporting stakeholders being companies, private and public institutions, national regulatory agency, and national control laboratories to implement non-animal testing opportunities whenever those are available. So you can actually get some of the information about the work that we are doing in the different regions and countries to the links that you can see here. You can also see all the events that we have been doing because our idea is really to interact with every stakeholders that is interested to move ahead this transformation. We are actually coming after a lot of important events and important changes that took place in the very few years. So there was a very important EDQM and European platform for alternative approaches to animal testing events in 2023. I hope that you are all familiar with this, but otherwise we will be happy to share with you the link where you can get access to a lot of interesting presentation and to a final meeting report. we wanted to build up from that events, and at the same time, we would also like to build from the feedback that was given to the World Health Organization from the NC3Rs report, where also we will hopefully see some updated guidelines on the pyrogenicity. So we really actually are glad for all the opportunity to discuss with many of you online, and we really hope to keep discussing with other stakeholders in new countries to really understand what are your complexities, issues, or your current experience in implementing this type of technology. I want to thank you all that participated to the pre-registration survey. That was for us extremely useful to understand your background, to understand your knowledge about the Monocyte Activation Test, and it helped us also. It helped us and also the speakers today to make their presentations and prepare for the feedback to give to you through the Q&A survey, the Q&A session and the final panel. We are actually seeing that we are truly a global community of stakeholders. So we see that there are a lot of people from many countries, from Europe, Latin America, North America, Asia, and Middle East joining us today. So we are extremely grateful of your time, and we really hope that we are going to give you an important experience and guide you through the change that you wish to implement and to stop relying on the Rabbit Pyrogenicity Test. With this, I'm actually happy to move to the workshop, and I would like actually to call for the first speaker of this workshop, which is Caroline Vipond from MHRA in the UK, that is going to give you some information about the work that she has been doing on developments, controls and standards for non-endotoxin reagents. Caroline, thank you for being with us. Please, the floor is yours. We can see your presentation.
Okay. Thank you, and good morning, good afternoon, good evening, whatever's relevant for you. It's just morning here for us. So yeah, I was asked to give a talk about some non-endotoxin pyrogen standard materials that we're making. I want to say straight away, this is a project I was quite involved in at the beginning, but I've not been so involved, and there are other people on the line, Trusha and Ingo, who have done a lot more of this work than myself. So I'm from the MHRA, the Medicines Regulatory Healthcare Products Agency in the UK, and the aims of the agency are to meet the highest standards for medicines. And part of the work we do specifically is in the National Institute of Biological Standards and Control, or as we formerly were, and we still make standards. So the standard materials that we make at the Institute are designed to act as a benchmark for medicines, and I'll talk some more about the different types of material that we work. But within the MHRA, there are a number of different groups which have all been brought together, and just to show you that this is the particular one where we sit. So why do we need these standard materials for biologicals? Biological standardization originated from a need to standardize variable bioassays, and the Monocyte Activation Test falls into this category of being a bioassay, where we're either testing on in vivo, in animal models, or you're testing on cells. And they can be difficult to measure because of the activity of the complex materials that we're actually exposing them to. So my experience is very much in the vaccine field. I worked very closely with a vaccine you will hear more about later on, and that's where I first became involved with this particular assay, and the ways that we could use the outputs from the cells to give us information about, in this case, safety. And the difference with a biological standard than a chemical standard is usually the unitage, where we actually use an international unit, which doesn't have a defined value like a microgram, but it tells you something about the actual activity of a product. And the other thing which we often do is talk about things in relative terms. So you will often measure your material against a biological standard, and it will have a relative value in comparison to the standard you're measuring it against. And these are very useful, particularly in things like batch release, where we're looking at batch to batch monitoring, and where you want to start to bring in some of the ideas of quality by design, where you're trying to show that your product is, for all intents purposes, identically made up as a previous product which was shown to be safe and effective in clinical trials. We make a number of different types of standards. So if we start at the top of the sort of hierarchy, this is the international standards, and these are WHO primary reference materials, and these have a defined unitage, as I said, often in international units. And a material that we will talk about more, I'm sure, over the day and also in my talk, is the international standard for endotoxin. And currently we're on the 3rd international standard, and this has been given a value of 10,000 international units per vial. So that's very typical, and that would be the highest order standard from which we would expect any value for endotoxin to be derived. The next type of material are international reference reagents, and these are also WHO reference materials. The difference with these is it's an arbitrary unitage which is usually assigned just as an interim reference material, and they're usually replaced by an international standard if that's deemed to be the right way to go. We then have working standards. Now these are validated against the primary standards, and they can be used more for routine day-to-day testing. So for our international standards, we would expect people to actually use those to assign a unitage to their in-house standards. So to try to preserve that material, this is the reason that we ask people to try to assign a unitage from our standard to theirs. But we are looking at this, and sometimes it may actually be more appropriate for people to use even our international standards in their day-to-day assays. And finally, we have a category of non-WHO standards, and we give these a status of an NIBSC standard. And these materials still undergo a lot of characterization and ensuring that they are fit for purpose, but sometimes it may be that the development of medicines are moving quite quickly, and we haven't quite decided where we need to put the material. So that is the other choice that we can have. And I think that, as I sort of mentioned throughout this slide, the main thing to think is that the status of a standard can change. You generally think that your international reference reagents might become international standards, but there can be cases where we may demote an international standard if there's a new one which we feel would be better. But just to say, it's a moving picture, whilst also having the highest order. And there are lots. I'll move on, sorry, and actually move on and talk about the Monocyte Activation Test. And I'm sure there's going to be some more introduction to this, and we'll probably see lots of these pictures. But I think I just wanted to get down to what are the basics of this bioassay that we are trying to understand. And what are we trying to get out of it? What is the end result that we are looking for? And it's all linked to whether or not our medicine, like I say, often when I'm thinking about vaccine, is likely to cause a pyrogenic response. And a pyrogenic response is it's denoted by having a fever, and these can be fairly serious, particularly in infants, where a very high fever can lead to a febrile response. And in very extreme cases, too much of a pyrogenic material can actually lead to organ failure and death. So there can be an outcome which is the worst that you could have. And what we try to do to replace what the Rabbit Pyrogen test as well was to think of really what's that measuring and what do we want to measure. And if we have a look at the toll-like receptors, which are actually sensing different pyrogens which enter into our body, be it from medicines, often from infectious diseases, we can start to have a look at which, what's really important for measuring this pyrogenic response. And from that point of it's what we've been looking at over the years in particular are these three pro-inflammatory cytokines, interleukin-1, interleukin-6 and TNF-alpha, and they're all known to be involved in inducing fever. And essentially you can have different pyrogens which may activate your different toll-like receptors. This green arrow represents a massive network of different intracellular signaling, but the outcome is the upregulation of these pro-inflammatory cytokines along with other things, and we can actually measure these, the output of which is often measured using ELISA. And I now wanted to have a think about we have as I've described, the different sorts of standards that we make. We know that what we want to have a look in the monocytes is this response to the toll-like receptors. And if we look at the pharmacopoeia chapters in the European Pharmacopoeia, there are particular things which stood out that we really wanted to have a think about. And first of all, that the test detects endotoxins and non-endotoxin contaminants. I think that's a very important part of this. And then to move to what do we need to do to validate our assay to show that we've got non-endotoxin and endotoxin, the ability to measure both of those. And the preparatory testing is suggested that you include at least 2 non-endotoxin ligands. And here in this part of the Pharmacopoeia 2.6.30, the Monocyte Activation Test, we have peptidoglycans, lipotenic acids, synthetic bacterial lipoproteins and flagellin as suggestions. And again, at the end of this, it says the test should ensure that at least TLR4 and 2 other TLR ligands are stimulated. If we have a look at the another part of the European Pharmacopoeia, and this is quite a new chapter, and this is to look at vaccines which are inherently pyrogenic. Again, if we look at the validation that's required for inherently pyrogenic components, it's suggesting that you need to check that your cells are able to detect the non-endotoxin pyrogens. And here there's a suggestion of whether if stimulating TLR2, TLR2/1,6 and TLR5. So I think it's trying to think what that means. So first of all, we've got to show that we can stimulate TLR4, and we know that endotoxin is the stimulator for this toll-like receptor. And as I said, we have available a standard in our catalogue which can be used to do this. And this is actually the 3rd international standard. I want to say that its value of 10,000 international units per vial were determined by the bacterial endotoxin test using the Lau method. So it is different from what you will get as a MAT readout, and that has to be thought of, but it's a TLR4 receptor. But in the Monocyte Activation Test, you can use this to qualify that you can get a TLR4 response in your cells. So just as a very basic kind of quality control test that the cells that you have are able to react to it. You can also use it to quantify the amount of endotoxin in a sample. But just to note that this will require a product-specific validation, and in particular, you need to show that the product that you're measuring runs parallel to the endotoxin curve. And you can use this to then define an endotoxin equivalent value. The materials we want to make are the new non-endotoxin pyrogens, and the TLRs we want to look at stimulating are going to be the TLR2, TLR5 and TLR7 and 8. And I just want to say that this is a project which is being run with EDQM, and in collaboration particularly with the Paul Ehrlich Institute, who have done a lot of work at helping us to identify suitable candidates for this assay. And again, I think we want to really sort of talk about what will the use of these materials be? How do you use them? And initially, they're really identified to qualify the cells. So they're a positive control. So this is the first step with these materials, is that people will be able to use them to satisfy those particular parts of the pharmacopoeia, which I just discussed, where you can show that the assay that you have is able to pick up non-endotoxin pyrogens as well as the endotoxin. I've said these will have no unitage. We haven't completely decided on how to take this, but this is how we envisage them in the first step, is that they won't have a unitage. So you will just know that if you use them, we will give a guideline dilution that should be used, so that you can see if your cells are able to pick up these other two pyrogens. And the other thing that we won't have is a limit which equates to a pyrogenic response. And this is something which is quite different. I think there's a lot of data on the back to the endotoxin, on the amount of endotoxin that's acceptable to have. So you can actually, when you have your specifications for your product, you can go with a number which you have faith in, that if your product actually contains less endotoxin than that, it should be safe. At the moment, we're not going to be able to give the same kind of values with these non-endotoxin pyrogen reference materials. So just to go back to the first slide and add a little bit more to it on the toll-like receptors that we've actually decided to look at. So as I said, we've got the endotoxin, which stimulates toll-like receptor 4 already. So the other one is the toll-like receptor 2, toll-like receptor 1 complex. And for this, we've had a look at material PAM3 cis lyse 4, and this is a synthetic triacylated lipoprotein, and mimics gram-positive type responses. The next one we've been having a look at is for TLR5, and this is FLAST, which is actually an acronym for Salmonella typhimurium flagellum. And finally, resiquimoid, which is a TLR7, TLR8 agonist. So one of the toll-like receptors which actually sit internally within the monocyte. And again, this is a well-known upregulator of these toll-like receptors, and has been used as an antiviral and also anti-tumor therapy. So there is quite a lot known about this this molecule as well, and its role as a toll-like receptor. So now to jump back to the process of making a WHO reference reagent or standards material. So we take our project to a committee who of experts, the Expert Committee for Biological Standardization, who will either approve or not approve the proposed material. And actually ourselves and Paul Ehrlich have taken different projects to the WHO, and they are supportive of us developing these and housing materials under the auspices of the WHO name. We then, once we've sourced the material, we work out process development, we have a definitive large-scale fill, we run a collaborative study, and that can involve laboratories spread around the world from as many labs as we can, really. And once we have the data from the participants, this is analysed by our expert statisticians at MHRA, who have many years of experience of working with the type of data that's used for reference materials. We create a report, and this material will be established again by the Expert Committee for Biological Standardization. And this timeline can vary greatly depending on many factors. But just to use that timeline as my basis, and I just call out to Paul, who made this very nice slide, to have a look at where we are with the materials that we've been developing. So for the PAM3 CISK4 and the R848, the project was endorsed, as I said, by WHO. We've sourced the material so that these are commercially available, which gives us an ease to replace them in the future, which is always something that we want to consider. We've done a lot on the process development, and this is we freeze dry the materials, and sometimes excipients have to be added because we need to make sure that the cake that's formed after lyophilization is stable, so that we can ship them around the world and be confident with their stability. So we've done small scale fills, and we've done some stability studies, and are very happy now with the material we have and the formulation we have and the freeze drying we have. There's been, and I'll show you some slides, also quite a lot of characterization of the materials that have been done both at the labs at MHRA and at Paul Ehrlich. And we're at the stage now where we're ready for the definitive fill, and this is planned imminently in the next 6 months or so. And once you've had a definitive fill, there are a number of criteria that we have to fulfill for WH materials just to ensure that that process has taken place correctly. And these are really all to do with making sure that we have a stable product that you can use to benchmark your assays against. And just to sort of say a call out, when we're also planning the collaborative study, and some of these things that I've mentioned, like unitage or how they might be used, should hopefully be answered within these collaborative studies. And I'd like to say, if there's anybody who'd be interested in participating in this study, we're always really keen to have more participants, and please get in touch with us. And also, if you just have any comments on how you think that we may need to do to make these the most useful materials and the most appropriate materials, please do get in touch. So we're in a good place with these, and we should have them ready to go fairly soon, hopefully in a timely way, so that when the European Pharmacopoeia removes the rabbit tests, that we have got these materials ready for people to use. Unfortunately, the story is not quite the same for the flagellin. We've found commercially available materials, but we've had quite an issue with stability. So at the moment, we're reassessing what we should do with that. But our aim is that we still get a TLR5 agonist in the panel. So quickly to move on, because I'm wary that my time is running out, onto some of the work we have done. We have just to show that we've shown that we can generate these nice dose response curves with the materials, and also we've used some reporter cell lines. So these are TLR2 reporters here, which have only got the toll-like receptor 2 versus some null cell lines, and as you can see, we've shown a nice response, and we will expand that study to ensure the specificity is right for those materials that we're providing you with. And we've also run a LAL test on all of these to ensure there's no contaminating endotoxin in there, which there isn't. We've also had a look here at some spiking experiments, and you can see the slight different behaviours with the PAM3 CISK4 and the R848, but we're seeing the types of things that we would expect here, and showing that we haven't got endotoxin contamination in them from the beginning. And some really nice work here that Paul Ehrlich have also done, which is to just try and give a comparison of, and this is the R848, of where you would expect the pyrogenic response as measured by IL-6 here to compare with the endotoxin, and where you would get these high fever rates. So as I said in the other slide, that's where we're up to. We're really looking at formulating the collaborative study and working out how to go forward. And as I said, if you would like to get in touch with this, my colleague who will be leading the collaborative study is Trusha Desai. So please, she's asked if you could email her directly. She'd be very happy to have your input. And that we're also going to continue to develop this TLR5 material. So thank you very much for your time, and thank you to the people who've done so much work on this, particularly in Ingo's lab, and Trusha and our colleague Kata, who's currently doing very important work on maternity leave. Thank you.
Thank you, Carolina. Thank you very much. I don't see any question at the moment, but please, I invite the attendees to just put their question in the chat. We can, in the Q&A box, we can definitely take it up online during the panel. And in the interest of time, thank you, Caroline, for being with us. See you later in the panel. I would like to move to the first session of this workshop that is dedicated to the blood-derived products. The first speaker is Dr. No from the Ministry of Food and Drug Safety, that is actually going to present the work that is ongoing in South Korea. I'm just going to bring up his slide. Hope that you can see them. Dr. No, the floor is yours.
Good afternoon, everyone. I am Hang-sik Ro, Deputy Director at the National Institute of Food and Drug Safety Evaluation under the Ministry of Food and Drug Safety in Korea. I appreciate the opportunity to speak at the AFSA workshop today. My presentation will focus on the research our institution conducting on alternative animal testing methods for plasma-derived medicinal products. Next, please. In recent years, the 3Rs principle has gained prominence due to growing ethical concerns regarding animal testing. The MFDS in Korea is committed to developing alternative testing methods that are crucial for drug development and quality control. We are also collaborating with organizations like the OECD and ISO to establish testing methods and guidelines. Next, please. At the Blood Product Division, where I work, we oversee the quality control and national lot release of plasma-derived medicinal products. Next, please. In Korea, there are currently 33 PDMP and Rabbit Pyrogen tests are conducted on 28 of these products. Additionally, mice and rabbits are used in potency tests for human anti-tetanus immunoglobulin and mammut antivenom. To minimize the use of laboratory animals, extensive research is being conducted to develop alternative testing methods. Next, please. For the potency test of human anti-tetanus immunoglobulin, we currently use an in vivo toxin neutralization method involving mice. However, we are actively working on an in vitro test method using ELISA as a replacement. Next, please. Another key area of our research is the potency assay for antivenom product. We are developing an enzyme immunoassay to replace the current method that uses mice. Next, please. Pyrogen testing is mandatory for quality control across pharmaceuticals, medicinal products, and medical device. As the market for pyrogen test grows, the RAL test has been considered as a potential alternative. although it has limitation. It cannot detect gram-positive bacteria or fungi. Next, please. To address these limitations, we are working on the monocyte activation test. MET offers higher sensitivity and accuracy than the Rabbit Pyrogen test. It is not species dependent, has no ethical concerns, and can detect all types of pyrogens, including endotoxins. In 2016, we conducted a foundational study using rabbit monocyte to develop MET. Since this year, we have been conducting follow-up research using human monocyte cell line and peripheral blood mononuclear cells. Dr. Cha, who leads this research, will provide more details after my presentation. Next, please. In addition to research, the MFDS has revised related regulations. In 2022, we eliminated the abnormal toxicity test, and in 2023, we introduced the recombinant factor C test. Moving forward, we plan to remove the anti-hemorrhagic test for mammouth antivenom potency and incorporate MET and histamine testing using LCMS. Next, please. This concludes my presentation. Dr. Cha will now provide a more detailed explanation of the MET research conducted in Korea. Let me introduce Dr. Cha, the team leader of the New Drug Development Center at the Daegu Gyeongbuk Medical Innovation Foundation. Thank you.
Thank you, Dr. No, and Dr. Cha, the presentation is now up. Feel free to start. Thank you.
Okay, very nice meeting you guys, and I appreciate for nice introduction. So my name is Bok-sik Cha. I'm senior researcher at New Drug Development Center. So today I'm gonna share with you guys with the advancing pyrogen test, the role of the monocyte activation test. Next, please. So as we all know that over the past decade, animal testing has been essential for the field of research and development across the industry, including the food and pharmaceuticals, as well as for the safety evaluation. Consequently, number of the animal use experiment has been increased, leading to sacrifice millions, even billions of the animals die, as you can see. With the growing ethical concerning about the animal use, there has been global movement toward a more humane and ethical research practice, emphasizing the 3R principles. Since the 1990, regulation on animal testing have been established globally, including the Europe, Asia, and United States. Notably, the US announced a plan to completely ban the animal testing by 2035. So it's very obvious that it is expected that the animal testing will decline, driven by the regulatory and ethical pressure. So this demand is clearly reflect the growth of the alternative toxicology testing market, which is expanding from the 12 billion to the 15 billion by 2033. So this global trend in South Korea, under the leadership of the Ministry of the Food and Drug Safety, has already established various alternative testing methods and guidelines. And among this initiative, this project is focused on the establishing and validating alternative test methods for the monocyte activating test. Next, please. So traditionally, after intravenous injection of the substance like a saline, some patients have a fever response, and this was later identified to be caused by the bacteria-derived pyrogenic substance. Consequently, testing for pyrogenic substance in pharmaceutical, medical supply, and medical device has become standard practice, leading to the growth of this market segment. So the detection of the pyrogen was conducted using rabbit, but as I mentioned, due to the increasing the animal right and concern, the use of rabbits has been declined. There is also LAL test, which utilize a horseshoe crab immune system. However, it fails to detect a certain pyrogen like a non-endotoxin gram-positive bacteria or fungi. So to overcome this limitation, the monocyte activation test was developed. This test realized the fact that pyrogen, including the endotoxin and non-endotoxin, binding to the toll-like receptor, which is expressed in monocyte, triggering the secretion of the several cytokines such as IL-1 beta, IL-6, and TNF. Using ELISA, the secreted cytokine can be measured. It provides a sensitive and accurate method compared to the rabbit pyrogen test. There is no variation, do not have any ethical concern, so it can detect all the pyrogen material and reflect in vivo condition. So therefore, numerous institutions now recommend using the MAT assay as a replacement for the traditional rabbit pyrogen test. Next, please. So various alternative test method has been developed to replace the RPT, for example, LAL, factor C, and MAT assay. Each assay has a unique application and sensitive level. However, LAL still has an ethical issue because it uses horseshoe crab. In addition, LAL and factor C assay are not sensitive to non-endotoxin pyrogen, such as virus and yeast. In contrast, MAT use human immune cells that react to a wide range of the pyrogen, both endotoxin and non-endotoxin. MAT is thus more aligned with the human fever response and can provide more comprehensive assessment of the pyrogenic risk, not only in the pharmaceutical product, but also biological and medical device and cell therapies. So this is why we try to establish the MAT assay. Next, please. The monocyte MAT assay utilized the principle of the toll-like receptor signaling to detect the pyrogen, including the endotoxins such as LPS and non-endotoxin pyrogen such as flagellin and other molecules. Toll-like receptor play a key role in immune system. They are expressed in immune cell like a macrophage and dendritic cell, where they recognize and binding to specific components of the pathogen. We are highlighting an issue that there is no standard positive control material for non-endotoxin pyrogen. The diversity of the non-endotoxin pyrogen makes the standardization of a complex change. They can originate from various sources such as bacteria, fungi, or even host-derived molecule. Each show different property that can trigger immune response in various ways. So to address this issue, more comprehensive characterizing and validation of the potential control material are required. Next, please. So in summary, we aim to establish and validate the monocyte activation test using human monocyte cell line and human PBMs. Our plan is to develop methods capable of detecting both endotoxin and non-endotoxin. Furthermore, we intend to explore potential of the replacing traditional methods such as RPT and LAL test. Next, please. So to set up the MAT, we list up the parameters cover the monocyte activation test, such as replication and limited of the detection and accuracy, precision, selective, and so on. Next, please. So we performed the MAT assay using the standard endotoxin and LPS. So due to the time limit, I'm not going to show all the data today, but the experiment was conducting using the 4 type of the monocytes and PBMC. We conducted the experiment by the varying the cell numbers for each cell lines and adjusting the endotoxin treatment time. In addition, we utilized the ELISA assay measuring the interleukin-6, interleukin-1 beta, and TNF-alpha. We optimized the MAT through the various sample dilation ranging from the 1 to 2 to 1 to 16. Next, please. And also we collect the non-endotoxin substance capable of the activating various type of the TLR receptor and conducting the test using the 10 different NEP. Just like an endotoxin test, we the experiment were conducting using the PBMC and 4 different monotype cell line, which I'm not going to show today. Next, please. As a result, we observed a good response to the NEPs in THP1 and HL60 cell line. However, not all NEPs show the reactivity. Among them, NEPs that activate the TLR 1, 2, 5, 6, 7, and 8 exhibit the best responsiveness. Additionally, it was observed that the response of the IL-1 beta was significantly higher compared to the IL-6. Next, please. Among the 10 NEP, we choose the 4 NEP, PAM3, HKS, flagellin, and CL075. We treat those NEPs in PBMC, and interestingly, while IL-1 beta show better responsive than IL-6 in monocyte cell line, however, in the case of the PBMC, IL-6 tend to be exhibit more strong response. Next, please. So finally, we're going to establish the MAT assay. A future experiment will focus following the key points. The first, collection and exchange the information. The second, we establish the monocyte activation test method and also the analysis of the monocytic phenotype change. And also investigating of the mechanism of the action of the pyrogen, and also the develop and establish in-house assay validation of the test. And we're gonna verification of the licensed plasma fractionation product and research on the monocyte activation mechanism and function. And last, we're gonna verification and compare with the Rabbit Pyrogen test. Next, please. So with that, I really appreciate to listening my presentation, and I'm going to finish this presentation. Thank you.
Thank you very much, Dr. Novo and Dr. Chang. a pleasure to having you. Let's see if we have some quick questions. Okay, so we can take one, if it's okay with you. There is a question about why you decided to switch back from the human cells from rabbits. It was a matter of cost or availability?
So you're asking why we choose a PBMC instead of the rabbit part, PBMC?
Why you went from the human cells to the rabbit cells at the beginning, and now that you make the switch again, what are the reasons for your choices and your change?
So the first we using the human cell line to address the NEP responsiveness. So which NEP will more sensitive to activating the human monocytes. And then we select those NEP to address the question in PBMs, in human PBMC, what will be the best choice of the NEP?
Thank you very much, Dr. Chow. Very appreciated. So we hope that you and Dr. No can stay as long as you can with us today. It has been a pleasure having you and learning about the work that you have been doing in South Korea about the implementation of the MIT. We look forward to collaborate with you in the future. I would like then to move to the other side of the world with the presentation from Deborah Ferrarin from the Butantan Foundation. Welcome, Deborah, and feel free to share your screen, and the floor is yours. Thank you.
Thank you so much, Lara, and thank you so much, you all, to be here today. I'm Deborah, and I'm a Senior Research and Analytical Development Analyst here at Instituto Butantan. And today I'll talk to you a little bit about our experience with MAT implementation with Sarah. So just to give you some context, I really want to share with you a little bit of our history. So in 1901, after a bubonic plague outbreak here in Brazil, the Instituto Butantan developed the first serum for this disease. Since then, we developed many types of serum and vaccines here. For example, the dengue vaccines, which we currently work on these days. And today we are the largest producer of sera and vaccines in Latin America. And each year we produce 506,000 vials of serum for Ministry of Health, which distribute them for free through our public health system. And among this number, we have 13 types of sera, and which we divide in 3 groups: antivenom, antitoxin, and antiviral. For realize this sera here at Butantan, we use the pyrogen test in rabbits. In 2022, for example, we used 306 rabbits for release the drug product. And in 2023, we used 651 rabbits for release the drug product. However, in October 2019, our National Council for the Control of Animal Experimentation recognized the MAT as a valid method for detection pyrogens in injectable product, and give us a period of 5 years, which ends in October of this year, to replace the original method by the alternative method. And with this movement, our Brazilian pharmacopoeia is being updated to include MAT in the chapter about pyrogen tests. And this revision is currently open for public comments now, and the final version is expected to be published in December this year. And our team have been looking for alternative methods to replace testing in all animals since a long time ago. And especially for pyrogen tests in rabbits, we start with a commercial kit with whole blood at the pest, but we did not follow through with it. And in 2017, we were part of a national research group to explore other methods to pyrogen tests in rabbits. But since 2022, we use a commercial kit with PBMC, and we validated, and the results we really want to share with you today. So after defining some MAT parameters, we move on to the validation. We divide it in two parts. In the first part, we validate the analytical methodology, and the second part was the suitability of the method for sera. In both cases, we followed as described in European Pharmacopoeia and in our Brazilian validation guide. So for the first part of the validation, we analyze some parameters like the selectivity, the limit of detection, the endotoxin standard curve, and the accuracy. For the selectivity, we tested different concentrations of endotoxin and non-endotoxin, with at least one of these concentrations for each pyrogen must be capable
Apologies, it seems that we lost Deborah. Give us a second to see if we can get her back online. Let's give her a minute to see if she can reconnect. Apologies. So while we maybe try to have Deborah back online, I wonder if we can take a couple of questions. Let me see if Dr. Cham, are you still with us online? There might be a question for you. Dr. Cha? Okay, let's see. Okay, so otherwise there is another question that maybe we can take in general about what is your experience, and I would like perhaps the other speaker to jump in. What is your experience between the monocyte cell-like and the PBMC? Which is the better for the MIT? Yeah, based on my experience and understanding, it really depends on the product. So you really need to try what is the best platform. But I'm happy to welcome the other speakers to comment on that while we wait to have Deborah back.
Laura, can you hear me?
Yes, I can, Ingo. Thank you.
Yeah, well, actually, like Laura said, it depends a little on your needs. So even if you ask people from the same pharmaceutical company, those who are doing research and development, they may like to work with the primary cells because they like to see the donor variation, because that's something that drug later has to cope with on the market. On the contrary, the people who later do the batch release, they do not like assays with donor to donor variation. So they might like more to have a kind of a monocytic cell line. Both would work in both versions, but I think the monocytic cell lines might give more stability. And if you control a known pyrogen, like in these pyrogenic vaccines, also there the monocytic cell lines might have a benefit, but both work fine.
Thank you, Ingo. Someone else wants to comment? Otherwise, I guess that's while we are trying to reach out to Deborah. Yes, please, Tonga. Hello.
Hi, Laura. Actually, I just want to echo what Ingo just said. I think this is also with our experience. As we all know, mass assay as a cell-based assay is still very, very much variable as for QC test. And if we could use cell line and manage the cell line using a banking system, and it's going to reduce the variability. I think from that point of view, it's a better option if it is for QC test. Okay, over.
Thank you, Tonga. Thank you for your comments. Very appreciated. Someone else? Otherwise, while we are trying to reach out to Deborah, I will probably propose that you move to the next presentation from Rolando. And then we will try to, we have Deborah back.
Okay.
I'm here, Laura. I'm so sorry.
Don't worry.
She's back.
Yes, thank you. Don't worry,
Thank
Deborah. I can share your slide if it's easier from your side in case your connection is not very stable.
Yeah, I appreciate that. Thank you so much. And I'm so sorry for this issue.
Oh no, don't worry. That's the way it is when we are all from online from our normal place. So give me a second. Let me know when you see your slides. Can you see them?
I can see. Yes. Could you go ahead, please? Yes. One more, and one more, one more, and one more. And after the validation for a few slides ahead. Next, 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 and yeah, here. So guys, I'm so sorry for this issue. And as I said, we evaluate in your validation the selectivity. And for that, we analyze different
We still have some issue with Deborah, I'm afraid. So, okay, we will resume with Deborah afterwards. So, Rolando, sorry, if you are ready, I guess that's probably that we move to your presentation while we try to see how we can help Deborah with that presentation. Give me a second that I bring up your presentation.
Okay.
And let me know if you can see it.
Okay. Yes, I can see it.
Perfect. Thank you.
So, okay, should I start?
Yes, please, Rolando.
Okay.
Thank you.
Okay.
Thank you.
Okay. So sorry to hear that Deborah couldn't finish, but maybe later. Okay. Good day. First, I would like to thank Laura for her kind invitation to this meeting and for giving us the opportunity to present our experience, which is in this case related with the using of MIT for assessing pyrogenicity in human seroalbumin for parental use. This study was conducted thanks to an international research project funded by the BMBF here in Germany between my institute in Cuba, the Center for Pharmaceutical Research and Development, the Paul Ehrlich Institute, and the University of Constance in early 2000s. The details of this work or this presentation can be found in the paper. You see the reference below that was published in Altex in 2011. It has been some time, I know, maybe long time, but hopefully we expect that the relevance of the topic remains. Next, please. Okay. In addition to implementing MIT in Cuba, as part of the joint project, we were interested in conducting a sort of comparative study or research about the performance of MIT in relevant samples. And eventually, and hopefully, those results could be used for a collaborative research paper. You know, it is important for project outcomes. From the very beginning, we considered that biopharmaceuticals, particularly therapeutic proteins, were one of the most exciting samples for the evaluation of MIT. After several consideration, brainstorming, discussion, we decided that human seroalbumin could be the best candidate in our hands due to the reason we mentioned here. First, we had access to blood products manufactured in Cuba, both pyrogenic and non-pyrogenic by RPT. There's real case samples. I would like to mention that there is still interest in implementing MIT there for safety reasons, but also for economical reasons. The other topic, very important, is that albumin can bind LPS. And to our eye, it is probably the first relevant example of protein masking of LPS. In addition, there were numerous various scientific reports related to the pyrogenicity of albumin, comparing human fever response with versus rabbits fever response, rabbits versus LIL, MIT, etc. We have noticed conflicting results and in the literature, and therefore it arises like as appealing or interesting research topic. At that time, the main pharmacopoeias, except the Japanese pharmacopoeia, indicated the rabbit pyrogen test for evaluating pyrogens in parental human albumin. And last but not least, pyroalbumin is one of the best-selling biopharmaceutical products worldwide. We recently found a paper published in 2023 analyzing the biopharmaceutical market in China, and albumin was the second biopharmaceutical on sales in 2022. Interestingly, immunoglobulins were the third. Next, please. Next, please. Hola.
Can you see it, Rolando?
Yeah, but I see next slide, please.
Okay, I go next.
Okay, sorry. Okay, in this study, we compare the performance of conventional and endotoxin specific LIL, the rabbit pyrogen test, no, before, the previous one. Please, the previous one. The previous one.
So, Rolando, let me see. What are you seeing?
Yeah, I am seeing the table. This one. Okay, this one.
Okay, okay.
So, okay. So I was explaining that here the overall aim of the study was to compare the performance of endotoxin specific and conventional LIL the Rabbit Pyrogen Test, and the Monocyte Activation Test using fresh human whole blood and IL-6 and IL-1 beta as readouts on 16 batches of human, 20% human seroalbumin for parenteral use. Next, please. Next, please.
Can you see it has changed from your hand?
I can see in.
Okay. I am on the interference test.
Yeah, now is the next slide.
Okay.
What I can see. Okay, here. Okay, okay. Thank you. As a typical, stated for LIL, we first conduct an interference assay on 10 batches of albumin to find the dilution free of interference. It is also called product characterization. Here, we found that the endotoxin recovery was above 200% at 1 to 10 dilution in half of the batches. You can see the red rectangle in the left column. Unfortunately, I cannot use the point. Such an enhancing response that you can notice there could indicate the presence of glucans in the sample. We knew that the albumin production process in Cuba employs cellulose-based filters, which are known to release glucans into the samples. Therefore, we then proceed to analyze the same samples using the endotoxin-specific LIL by including glucoshield buffer from ACC to inhibit the well-known factor G glucan pathway. As you can see there, the endotoxin content in the right part of the table, the endotoxin content estimated in the samples decreased. However, the endotoxin recovery also dropped below 50% in most of the samples. What is really notable here, as you will see later, is that the only samples showing more than 50% of endotoxin spike recovery were those that failed the Rabbit Pyrogen Test. Dr. Donash Hastings at the National Institute of Health, who was one of the pioneers in the implementation of LIL assay in the pharma industry, he published the study in 1979. You can see there the citation, where he and his colleagues assessed the suitability of LIL for endotoxin control in over 50,000 batches of 20.5% human seroalbumin produced by US manufacturers. In this study, they suggest that albumin masks specific amounts of endotoxin until saturation occurs. It is highlighted in the square. So the LIL test was not able to fully detect the endotoxin spikes. In addition, the possible masking effect of albumin on endotoxins could explain why the no enhancing reaction to endotoxin spikes promoted by glucans was only found in a few dilutions in some batches. As most of you know, the enhancing reaction to endotoxin spikes, which is indicated by more than 200% of recovery of the spike, is a typical indication of glucan contamination. Therefore, this result may indirectly indicate that the LIL was not able to detect the endotoxin spikes in most batches or their dilutions. It was undetected, masked. In other words, there was no LIL response to the spike, and therefore no enhancing reaction was evident in most of those cases. Next, please. Next.
Has it changed?
Yeah, okay, perfect. So here we show partial conclusion concerning LIL or BAT. All 20% albumin batches were contaminated with significant amounts of glucans, which could go unnoticed by the typical enhancing reaction to endotoxin spikes, especially when endotoxin is undetected by LIL. This could lead to possible source of errors, and in In worst cases, false positives. However, research on out-of-specification results could indicate the presence of glucans, and the issue, or this issue, could be easily solved. Albumin may mask endotoxins to the LAR region, leading to failures in the recovery of the positive product controls of our spikes. The second point, the estimated stoichiometry found for albumin and LPS binding is 1 to 10 molar. Above this relationship, albumin becomes saturated, and the toxin is probably detected by LIL. Finally, the lab test in our result, in our study, was not suitable for assessing endotoxin content in human seroalbumin due to interference problems. Generally, the hot spike recovery was less than 50%. It's worth, we would like to mention that previous studies conducted by Japanese researchers in the 90s found that endotoxin determination by LIL in albumin correlates with the Rabbit Pyrogenicity Test. Moreover, they found that suitable spike recoveries using endotoxin-specific LIL. We know, in fact, that BAT was implemented in Japan since at least 2006 to assess endotoxin in albumin. However, we are not currently aware of the current status or regulatory status. We would like to mention that the control of pyrogens in blood products, particularly albumin, is still a hot topic to our view, and the scientific literature shows conflicting results. To our eyes, more comprehensive and harmonized interlaboratory studies involving samples from different companies may be interesting. Welcome. Next, please. Next. Okay, perfect. Here in this table, we show the result of albumin characterization using MIT and IL-1 beta and IL-6 as readouts. As you can see in the table below, surprisingly, we found that the IL-6 response was enhanced. The dilutions, the recovery was higher than 20 to 100%. We could not quantify the level of enhancement above 200% because we use a spike of 0.5 endotoxin unit per ml, and the maximum concentration in the standard curve was 1 endotoxin unit per ml. Such a limited dynamic range did not allow us to quantify an enhancing response above 200%. In this case, we suspect that like LIL, but through a completely different mechanism, this may be an enhancing effect of glucans from filters on the IL-6 response to endotoxins, since a similar situation or result was described earlier. Notice, please, that conversely to BAT or MIT IL-6, no enhancing effect was evident in the hot spikes when the IL-1 beta was used as readout. You can see the minimum valid dilution for IL-1 beta was 1 to 4. Even only one batch in the upper side of the table, the batch C, that have a very low endotoxin recovery, but all of them are the endotoxin recovery inside the suitable range between 50 and 200%. Okay, next, please. So in the following table, we summarize the comparative study. The samples were tested with endotoxin-specific BAT at 1 to 100 dilutions, only for comparative purpose. The sample for MIT were tested at their minimum valid dilution for each readout. Notice that the endotoxin here at the right, upper right, the endotoxin equivalent concentrations estimated using the IL-6 readout were generally higher than those obtained with IL-1 beta. There were in many cases with twice or more the endotoxin equivalent concentration was obtained with IL-6 readout regarding to IL-1 beta readout. In another sense, batch C, you can see there on the for the IL-6 in the second column. Okay, perfect. IL-6, the batch C failed the MIT IL-6, but passed the MIT IL-1 beta, the Rabbit Pyrogen test, and the LIL test. Then we found that 2 batches failed the MIT, both cytokines, the RPT, while 1 of them passed the LIL test. Okay. Finally, a third batch, I, the letter I, failed both LIL and RPT tests, but passes the MIT IL-6 and MIT IL-1 beta tests. If we take a close view to the result of MIT IL-1 beta, MIT IL-6, and endotoxin specific LIL, we will notice that the results were in the borderline of the endotoxin limit. If we could, or if we perform or conduct a statistical comparison, you will notice that the results are quite similar. The standard deviation is okay. Maybe there are not a statistical significant difference there, and maybe the results, since are in the borderline, are very or quite similar. Okay, next. Next, please.
Can you see it?
Yeah, perfect. Okay, thank you. Next, we would like to comment that we have recently learned that the Rabbit Pyrogen test is not longer mandatory for testing albumin for parents. Instead, it seems that the using of bacterial endotoxin test is encouraged. We try to illustrate a little bit with this warning symbol. You know, according to the USP, they said the albumin should be in the albumin monograph. No, they sent us to the chapter Biologics 1041, and in this charter is like a grade statement. So you can use Pyrogen test, you can use bacterial endotoxin test. Our consideration, we humbly think that we need clear indication for this case. In the past, were clearly stated that the Rabbit Pyrogen test has to be used. But now this is like a gray situation is also evident in the European Pharmacopoeia, where this is stated in the albumin monograph that it complies with the test for parents, or preferably and were justified and authorized with a validated in vitro test, such bacterial endotoxin tests. So this call our attention and for us drive us to the next and final slide. Next, please. Okay.
It's up.
Okay, so just thank you. Summarizing why we suggest considering MIT for testing albumin for parents. Okay, first, as we all know, resembles better the human fever reaction. But to our point of view, and more importantly, albumin binds to endotoxin, and they have a series of consequences. For instance, some authors include albumin as part of the LPS binding protein, similar to high-density lipoproteins and other proteins. Albumin's ability to bind LPS has been used for endotoxin removal. This system from Fresenius and all as in albumin has been also used to remove endotoxin from samples that interfere with MIT or BAT. Okay, albumin presents endotoxin to the immune system more efficiently, inducing a more significant response to the same concentration of free LPS. So in our eyes, in our humble consideration, albumin does not mask endotoxin to the human immune system. You can see our result about the measurement of IL-1 beta response found in this study, but also in other studies. The literature, the scientific literature describes that albumin can disaggregate LPS into more active aggregates for the human immune system, causing a stronger response. It is also known that albumin can present LPS to activate the toll-like receptor 4 IMT2 system without the participation of soluble CD14. All of these facts could be particularly relevant when considering the assessment of pyrogenicity of albumin in humans, which could also, as you may know, differ to the rabbit response. Other point important to consider is that albumin's biological half-life is 19 days. According to the scientific literature, in a healthy person, it will last around 16, 18 hours in the circulation, but it can go back to the circulation through the lymphatic vessel. In these cases, in cases of a patient which is health compromised, contaminated albumin could pose a challenging scenario when administered parentally. For instance, in the case of liver cirrhosis, where the degradation of albumin is impaired and where the degradation of LPS is also impaired. And finally, endotoxin bound to albumin can be really detected by whole blood MIT L1 beta, as we show in this study, but masked to LIL. So finally, considering the ability of albumin to bind and present LPS to the human immune system, we consider that MIT, we humbly consider it, according to our own result, that MIT is the most suitable assay to assess pyrogenicity in human in albumin formulation for parenteral use. Next, please. Thanks. Thank you for your attention.
Thank you very much for your presentation and for sharing your experience, Rolando. Very appreciated that. So let's see if we have some questions. Otherwise, I understood that Deborah is back online. No questions, but please do keep adding your question into the R&D. Hello, Deborah.
Hello, Laura. Yes, I'm so sorry again.
No,
I think now everything will work.
Okay, so do you want to try to back to your presentation? Oh,
yes,
great.
I can. Perfect.
Perfect, we can see it.
You can see? Yeah, perfect.
Thank you.
So,
and thank you, Ronaldo, for your presentation. So, Deborah, the floor is yours.
Yeah, thank you so much. And as I said before, so the first part of our validation, we evaluate the selectivity, where we tested different concentrations of endotoxin and non-endotoxin, and at least one of these concentrations for each pyrogen must be capable of induce PBMCs to produce IL-6 above the basal response. And as a result, we obtain at least one of these concentrations for each pyrogen was capable to induce the IL-C response above the basal response. And for the LOD, we performed as described in European Pharmacopoeia. And for this parameter, we set the acceptance criterion with the LOD must be less or equal to 0.016 endotoxin unit per ml. And as a result, we obtained the LOD in all assays was less or equal to 0.016 endotoxin unit per ml. For the endotoxin standard curve, specifically for the LPS standard curve, we proved the 4PL regression model was fit for purpose. In this case, we evaluate some criteria like the significance of parameters B, C, and D, the coefficient of determination, the homocelasticity of the model and the CV% between the replicates of the same concentration at the curve. And as a result, we obtain the significance of parameters B, C, and D with the T-student test. As well, we obtain a coefficient of determination greater than or equal to 0.975. And also, we obtain the homocelasticity of the curve. And also, we evaluate and prove that the coefficient of variation of each concentration was less than or equal to 30%. And for the accuracy, we evaluate the recovery of LPS in 3 different concentrations. In this case, we evaluate the low, the middle, and high concentration. And the recovery was calculated using this equation represented in the slide. And for acceptance criterion, we set the recovery must be with 50% and 200%. And as you can see in this graph, we obtain for the 3 concentrations of LPS recovery between 15 and 200% as we define for acceptance criteria. And finalized the first part of the validation, we move on for the second part we analyze the suitability of the method for sera. In this case, we evaluate the sera interference in the ELISA assay. As well, we analyze the sera interference in endotoxin and non-endotoxin detection and the intermediate precision. And since the matrix of all sera is the same, we choose one representative of each group to follow the tests. For the first parameter, the sera interference in the ELISA assay, we create a IL6 curve with the ELISA buffer and with each type of serum. And as an acceptance criterion, we define the coefficient of variation need to be less or equal than 20%. And as a result, we obtain the coefficient of variation between the curves less than or equal to 20%. As well, we analyze the angular coefficient between the curves, and the result was the curves were parallel, as you can see in this photo here. Just a representative photo of our assay. For the sera interference in endotoxin detection, each serum was tested at 3 dilutions, with and without the addition of LPS. And for that, we calculate the LPS recovery using this equation, representative at the slide. And for the acceptance criteria, we said the LPS recovery must to be between 50 and 200%. And as a result, the 3 sera had the LPS recovery between 50 and 200% in 3 independent assays. For the sera interference in endotoxin detection, the 3 types of serum was tested in just 1 dilution, and with and without the addition of HKSA. And for the HKSA recovery, we calculate this number with this equation, representative here at the slide. And for the acceptance criteria, we set the HKSA recovery must be between 50 and 200%. And as a result, we obtained the 3 sera had the HKSA recovery between 50 and 200% in all assays performed. And finally, for the intermediate precision, we evaluate the reproducibility of the method by 2 different operators. And as a result, we obtain the agreement between the sample results by the 2 different operators. And as a conclusion, the MAT can be considered validated and suitable for implementation in quality control routine for pyrogen analysis in sera. And just to give you just an estimated of time and costs we had since the analytical development and the suitable of the methodology. This is some numbers about the tests and analyst hours or investments, about consumables and our labor costs. And implementing a new method, it's always challenging, and we really want to share some challenge with you. When we start to use the MAT assay, define some parameters was a really challenge, especially when we need to analyze the regression model used for the LPS standard curve. Also, during the development, we found some interference with drug substance in the MAT. So we decided to test only bulk and drug product on this method. For drug substance, we will use the validate LAL and RFC tests as well. And last but not least, the other challenge is the finite specification for the product, especially for syrup, because we have different products with different dosages. So this is our research and analytical development team who has been working hard in many projects, and as well as alternative method to the use of animals in quality control routine. And I'm really proud to be part. And I want to especially thank you, Fabio Sato, for his dedication in the MAT implementation here at Instituto Butantan. And thank you so much for your attention. And I'm sorry again for all these issues. And please, if you have any question or if you want to discuss this further, you can contact us by email. Can you see it here? Oh, so thank you so much again.
Thank you very much, Deborah, and also for rushing.
No worry.
Very appreciated. We are a little bit tight with the time. So I would try to move to the next presentation. And sorry, Antti, for your question. If you can hold it a little bit later, then we will bring it up afterwards. Thank you, Deborah. I would like to the next presentation from Jacqueline Shepherd from the National Bioproduct Institute from South Africa. I'm going to bring up perfect. Thank you, Jacqueline. We can see your presentation, and the floor is yours. Thank you.
Thank you. Can you hear me?
Try to speak again.
Sorry, can you hear me now?
It's a little bit low, your volume.
Let me see if I can do anything.
Let's try again. Try to speak.
No, can you hear me now?
It's still a little bit low, but if you can speak out a little bit louder, I guess that's where we'll try to handle it. Thank you.
All right, is this okay now?
Yes, much better. Thank you.
Okay. All right. Good afternoon, everyone. My name is Jacqueline Shepherd. I'm a scientist in the product development team at the National Bioproducts Institute in South Africa. I'm looking forward to sharing the NBI MAT experience with you.
Sorry, Jacqueline, sorry to stop you, but then you went still a little bit lower.
So stop. Let me just pull this And now?
Doesn't change.
Doesn't change. Let me see. Perhaps if I... Just going to exit quickly.
Okay. Okay, then while we wait for Jacqueline to come back, I will try to move to the next presentation. Marike, are you with us? So then we save some time while we get back.
Yeah, I will
Thank you.
start sharing. Yeah. Can you see my screen?
That's wonderful. And thank you, Marike, for jumping in. So sorry,
So,
all. So we will try to get back with Jacqueline back. And we are moving to Marike's presentation that is going to share her experience again with the human albumin preparation. So Marike, thank you for your availability and for being with us today.
Yeah, so thank you very much, Laura. So I'm Marijke and I'm a senior scientist for Med Services at Sanquin Diagnostics, which is a part of the Dutch Blood Supply Foundation. And just as discussed earlier today, there are different MAT formats. So you can use whole blood, fresh PBMCs, cryopreserved PBMCs, cell lines, which all have their own advantages, disadvantages, and also different readouts. So you can have IL-6, IL-1 beta, and TNF-alpha, which were already discussed upon today. And nowadays you also have cell lines with reporter genes. So there, the TLR activations usually all combine into NF-kappa-B activation, and then they have NF-kappa-B luciferase to measure so that you can more quickly measure if there were pyrogens in your product. In addition, also there are differences if you use single or pooled donors or cell lines, because cell lines are more a single donor. Because, for example, it's known for TLR2 and TLR5 that there is a lot of genetic variation throughout the population. And with using multiple single donors or a pool of donors, you can at least catch the genetic variation. For example, it can be that one of the single donors or your cell line doesn't respond to one of the pyrogens as you would expect, because usually all people respond quite comparably to endotoxin, but to the non-endotoxin pyrogens, there's a lot of variation between everyone. We have seen it all during the COVID pandemic. Some people got ill and very ill, and others, they were even as asymptomatic. So you can cover that when you really think about your MAT assay and why you want to use it. At our laboratory, we use the cryopreserved pooled PBMCs from 4 donors, and these have a lot of advantages, at least in our eyes. So they are available on demand, so you don't need to draw fresh blood up front of your experiments, and you can obtain 300 to 500 files with identical cells. So you or your vendor can qualify these cells up front, and you know if they would respond as you expect before starting your MAT assay. So that's, for us at least, a big advantage. And they are more sensitive than the cell lines, in our hands at least. In addition, we use a pool of 4 different donors. So to take into account the donor variation, which I addressed just now. And the cryopreserved cells, they are stable for years. So in liquid nitrogen and for months at -80 or colder, but you really need to have it -80 and not -70. In addition, we use the IL-6 usually as a readout for our MAT, since it has a high sensitivity and a good correlation with fever in the human body. Sorry, sometimes I'm still recovering from my cold, so I need to cough sometimes. In the MAT, there are also serum factors necessary to provide essential proteins to the monocytes to activate them when they are exposed to the pyrogen. So, for example, you would need LPS binding protein, LBP, to bind to LPS and present it to the TLR4 receptor before it can be internalized. So you would need some serum factors, and at the start of the MAT at Sanquin, at least in the end of the 1980s, beginning 1990s, the groups used fetal bovine serum as a serum source, since that was then commonly used in the cell culture field. And therefore also the first MAT kits from Sanquin reagents, which is currently called Assange reagents, they also contained FBS. However, when we started services, our clients also asked if we could have a human serum based, so HS based MAT, in order to not use the cruelly obtained fetal bovine serum, and also to better reflect the human immune system, because you use human serum with the human cells. And of course, our body, also everything is human. However, there are differences when you use the different serum sources. And in the next slides, I will show some data which we have also published. So you can find it here. Is a reference to the paper if you want to refer to it or look into it more deeply. So here you can see the effect of fetal bovine serum, the blue line, and human serum, MAT. So it's the same cell, same readout, only a different serum source. And then the response to endotoxin. So on the x-axis, you can see the endotoxin concentration, and on the y-axis, the IL-6 response in optical density. And here you could see that with FBS, we had nice LPS curves, which had a high sensitivity, so of 0.02 endotoxin units per ml. And with human serum, the curve is shifted towards the right. So it's less sensitive for endotoxin. And then the sensitivity is 0.08. Does it work? Yeah. And so there's a somewhat lower reactivity towards endotoxin when you use the human serum-based MAT. However, the effect on non-endotoxin pyrogens is different. So if we looked at, we have selected several non-endotoxin pyrogens at which we wanted to look. And we found higher reactivity with human serum for the PAM3 CSK4, also for heat-killed Staphylococcus aureus, and for PGN. And as you can see, the red line, which is again the human serum based assay, that one is now more towards the left. And you can see it for heat-killed Staphylococcus aureus and for PGN. Other non-endotoxin pyrogens which we have tested showed comparable reactivity between FBS and human serum. So, there's maybe a trend for better human serum, but we indicate it as comparable. And R848, that was really comparable between the different assays. So to conclude at least the effect of the serum source, the use of human serum gave higher reactivity towards most of the tested non-endotoxin pyrogens. So to the PALM3, CSK4, the heat-killed Staphylococcus aureus, and peptidoglycan. And it had no difference with flagellin or R848. However, the use of human serum has a lower reactivity towards endotoxin compared to the FBS-based MAT. However, the MAT is still a pyrogen test, so it should detect all pyrogens. So therefore, we feel that a human serum-based MAT is in most cases a better option, since it also better reflects the human immune system and the reactivity towards non-endotoxin pyrogens is more sensitive. The differences which we see in endotoxin reactivity when you use FBS or human serum-based MAT, they can be caused by differences maybe in the LPS binding protein amount between fetal bovine serum and human serum, or as discussed also by Ronaldo, the differences in serum lipoprotein content. So LDL, VLDL can inactivate the LPS. Maybe there is presence of anti-LPS antibodies. We never found them yet, but it's still a hypothetical option which can cause these differences. But there's of course one downside of having a lower reactivity towards endotoxin, that there is a higher test sensitivity for the human serum-based assay. And therefore, you will get lower maximum valid dilutions because we know the formula to calculate the maximum valid dilution. So it's the CLC, which is equal to the ELCs, to the endotoxin limits, and divided by the test sensitivity. So if the test sensitivity has a higher value, your maximum allowable dilution, that will be lowered. So I will show you 2 slides later on the effect which it had, at least at our albumin testing. But why would we test albumins with MAT? So before, it was already indicated that MAT might be the better option because there are differences between the BET and the Rabbit Pyrogenicity Test and the MAT testing. And the research group of Sanquin has also indicated that and published some of that data. So they tested 4 albumin batches with BET and with a Rabbit Pyrogenicity Test and with MAT. And 3 batches were contaminated with RPT and also indicated to be contaminated with MAT. However, they all were missed with a bacterial endotoxin test. So therefore, albumin preparation, at least we showed also that there are a lot of TLR2 activators within or agonists within the batches, together with also some TLR4 activity. So it's a bit a combination. So a combined contamination within the albumin preparations, but due to the fact that they can contain non-endotoxin pyrogens, the BET is not appropriate to test albumin preparations because you will just miss them because the BET will only measure endotoxin and not the non-endotoxin pyrogens. So as just stated before, we at first used the FBS-based MAT in our laboratory for batch release testing of albumins. And here is some of the data which we have obtained during those years. So the CLC for 20% albumin preparation, at least here, is 1.3 endotoxin units. So per ml. And if you divide it by the sensitivity of the FPS-based MAT, it's at 65 times dilution per sample, which will result in a 130 times dilution in the well. The product-specific validations have then indicated which dilutions we can use. So the lowest dilutions where there is a acceptable endotoxin recovery between the 50 to 200%. And those dilutions were 12, 24, and 48 for the FPS-based MAT. And then we also found quite often enhancements in batches of albumin. Sometimes they are a bit lower. I don't know what is the difference, if there is then something with a new filter or an old filter, because we cannot look into that anymore. But all the batches could be tested and they are batch released with MAT. Then later on, for another client, we have switched to a human serum-based MAT, since now we also have human serum-based MAT available in our service package. And here you can see the effect that really on the dilutions, you cannot dilute as far as you could with FPS-based MAT. However, with product-specific validation, so this is 42 times maximum dilution per well, and these are also dilutions per well. There we could already indicate with the product-specific validations that the dilutions of 10, 20, and 40 were okay to use. During batch release testing, we also find again those higher endotoxin recoveries, and even sometimes really above measuring range. So therefore, there are asterisks here. So that's way above the 200%, but that is also in the plateau phase. So in the plateau phase, you cannot really correctly measure anymore the percentage, because there you're in the plateau phase, and a really small increase can have a big effect. And so the variability is higher there, so the precision is lower. So these data at least show that both serum-based MAT, so FPS, and human serum can be used for testing albumin, and that it depends on your registration dossier, which was accepted, of course, and the requirements of the client, which you can use. So at this stage, we usually prefer to start with human serum-based MATs, and then if there are difficulties or not valid results, then we will switch maybe serum source or do a different thing. At this stage, we have seen for other plasma-derived medicines that human serum is often a better fit for purpose in our hands with the samples which we have tested until now, because sometimes human serum-based, or often human serum-based MAT gives acceptable endotoxin recoveries, so within the 50 to 200% range, while with FBS we can have recoveries below 50%. So even though the maximum valid dilution is lower with human serum-based MAT, we often get better recoveries. So it can be worthwhile at least to see if your product doesn't work with MAT, if you can change your serum source. So to conclude, human serum-based MAT has a lower sensitivity towards endotoxin, but a higher sensitivity towards multiple non-endotoxin pyrogens. So, and it's also better resembling the human immune system, and it's much animal friendlier, of course, than the FBS-based serum, because human serum is just drawn from human donors and pooled and then used to test, or then used for in the MAT test. And for MAT testing of albumin preparations, both serum sources can be used. However, for other PDMPs, this can be different. So then I would like to thank some of my colleagues, of course, the MAT service team with a lot of technicians and the R&D team, and from Sanquin Research, especially Anja, who has helped us really a lot with the immunological details.
Thank you very much, Marieke, for your case study. Very interesting, the results on the use of human sera versus the FBS. That's very interesting because probably many organizations will have to take a decision whether to go and use one of the two. So I hope that your experience is going to be useful for them. Thank you, Marieke. We are probably going to take questions just at the end. So we move back to Jacqueline if she's back with us and hopefully with a much louder microphone. So Jacqueline, hope to see you and hear you loud and clear.
Hi, Laura. Can you hear me now?
Perfectly. Perfectly. Thank you.
Okay. I'm just going to share from my side. Just give me 1 second.
Of course. Thank you. Okay. All good here.
Okay.
Perfect.
All right. Okay. Thank you very much for the invitation to present at this workshop. My name is Jacqueline Skeepers. I'm a specialist scientist in cell biology at the National Bioproducts Institute in South Africa. I'm looking forward to sharing with you our NBI MAT experience, and I hope that it will provide some encouragement for those that are starting out on their MAT journey. So today I'm going to cover basically an introduction to NBI and its products. We will look at briefly at pyrogen testing at NBI, both past and present. I'll then move on to the development of the NBI MAT. We will look briefly at validation, implementation, and obviously some challenges that we have faced. So NBI is a private non-profit pharmaceutical company that manufactures plasma-derived medicinal products, or PDMPs. These are from fresh or frozen plasma of human origin using the process of cold ethanol fractionation. Our products are marketed, sold, and distributed to reach South African patients, as well as patients in neighboring African countries. The company's manufacturing site and head office are located in Pinetown in the province of KwaZulu-Natal, South Africa. If we look very briefly at our products, we can divide these into two groups, the pharmaceutical PDMPs, and then obviously the diagnostics. So in terms of the pharmaceuticals, we have a total of 13 therapeutic protein products of human origin. These include albumin solutions, specific immunoglobulins, coagulation factor 8, factor 9 complex, freeze-dried human plasma, and then obviously some normal immunoglobulins. And then in terms of the diagnostics, we make a rhesus typing test kit, and then monoclonal antibodies against hepatitis B, and then a range of antibodies against malaria. So all our NBI pharmaceutical products are registered with the South African Health Products Regulatory Authority, known as SAHPRA, and additionally, we follow a number of international regulatory requirements as shown on this slide. So if we move on now and look at a brief sort of timeline of MAT testing at NBI, the timeline begins really in 2015 when MAT product-specific validations were conducted by an external testing laboratory in Amsterdam on behalf of NBI. Once it was determined that MAT testing was compatible with the NBI products, approval was sought from our local regulator. Although I was not working at NBI at this time, I'm told that there were no issues in being granted approval to change from the Rabbit Pyrogen test to the MAT. The regulator was satisfied with the validation data, and this, together with an extensive literature review on the mat and its sensitivity, ensured a smooth transition. In 2019, NBI initiated work on the development of an in-house mat. Once the test had been developed and product-specific validations began in 2022, and in 2024, routine mat testing began at NBI. Currently, we are only testing a few of the NBI products as we await for final approval from our regulator. So at this point, you're probably wondering, why would we choose to develop our own mat when there are plenty of commercial kits available? So let's look at this from the South African perspective. So there are 4 primary reasons as to why we went for the in-house development route. First of all, cost. We assessed the cost of commercial kits and found them to be extremely expensive. We performed a costing exercise to determine whether the cost of developing our own assay made economic sense versus purchasing commercial kits. And it was very clear that from a cost-saving perspective, development of our own assay was the way to go. Secondly, delivery times. During the development stage of the NBI mat, we attempted to purchase commercial kits for comparison, and these are from well-known suppliers. Although these products are easily found on the international market, our local distributors were not familiar with them. And it took me approximately 12 months from my first inquiry until the product arrived at the laboratory. I admit that with time, should we have continued to purchase this kit, the timelines would have improved. However, for similar products, delivery times of 8 to 12 weeks are generally the norm for us in South Africa. This is quite challenging and requires extensive planning and safety stock to be in place should you be relying on these products on a daily basis. Thirdly, regulatory red tape. And here I'm referring to the import regulations should we have opted for the commercial kit. Because the mat kits contain human cells, whether they be cryopreserved PBMCs or the cell line, the Monocyte cell line, it becomes extremely difficult to import such products into South Africa. Import permits are required, and these are quite difficult to obtain, and the application process lengthy, just adding to an already long delivery time for the product itself. Often, the suppliers are not willing to apply for the import permit on behalf of the customer, leaving the customer with a rather daunting application process and ongoing follow-ups until the permit is issued. Although going the route of developing our own assay has not left us immune to this process, we still require import permits for some of the reagents that we use. However, we have at least found suppliers willing to handle the import of these reagents on our behalf. And then lastly, in-house capability. We already had the necessary technical skills and well-equipped laboratories for development and production of a mat kit for NBI use. So, although the development of our own mat may have not been the easiest or quickest route, it made sense for NBI in the South African context. So if we move on now to the actual development of the NBI mat. So we all know the mat is built on the principle that monocytes, when activated by pyrogens, produce cytokines or interleukins, and these can be detected in an immunological assay such as an ELISA. Therefore, there are 2 important aspects to consider when developing the assay. First of all, monocyte activation, i.e. your immunological response, and this speaks to your cell source. And then secondly, your ELISA and the detection of a specific interleukin. So I'm going to just look at these 2 separately, starting with the cell source. So when considering the monocyte source, there are various options. For example, it's whole blood or isolated monocytes such as PBMCs, or even using a monocytic cell line such as the Monocyte. Although the use of the cell line may seem easiest, there are several disadvantages to this, and also licensing and royalty fees, which we came across when we pursued this avenue. So in the end, we actually opted for producing our own cryopreserved PBMCs. So here we take a pool of 4 blood donations, and we are fortunate to have secured a group of NBI employees who are willing to donate blood for the mat. Additionally, we have a long-standing relationship with the South African National Blood Service, SANBS, who assist us with doing the actual blood draw. And we try to coincide the processing of a batch of PBMCs with the regular blood donation clinics that take place on site. PBMCs are isolated by density gradient using FICOL and are eventually stored in liquid nitrogen vapor phase. And thereafter, we begin the process of qualifying the PBMC pool for use in the mat. Obviously, we have faced a number of challenges when it came to preparing our own batches of cryopreserved PBMCs. Firstly, our first batch, because we lacked technical expertise in-house, our first PBMC batch was actually prepared by an external laboratory who are experts in PBMC isolation. However, for various reasons, this was not going to be something that we could pursue on an ongoing basis. So we realized that we needed to bring this capability in-house. Our staff were trained on PBMC isolation, and although we found the actual process of isolating the PBMCs not to be difficult, it is simply the scale on which it needs to be done and the time frame, which is normally 4 to 5 hours, that makes the process quite challenging and extremely labor intensive. We also have limitations in terms of centrifugation capacity, and this has restricted us on the volume of blood that we can actually process in the ideal time period. If we look now at how we go about qualifying the cryopreserved PBMCs, so like I said, PBMCs isolated from individual donors are compared with the pool, and we are pooling an equivalent number of cells from each of the 4 donors. And preparing a pool overcomes donor variability. Dose response curves are generated using the endotoxin standard and must meet the requirements stipulated in the MAT monograph. Further to this, we generate dose response curves for non-endotoxin pyrogens such as peptidoglycan and flagellin. So moving on now to the interleukin detection, in other words, the ELISA. So human immune cells such as monocytes respond to endotoxins and NEPs by production of pro-inflammatory cytokines like interleukin-6, interleukin-1 beta, or tumor necrosis factor. Literature has indicated that IL-6 has higher sensitivity, and so we settled on a human IL-6 ELISA. Although we have the necessary in-house capability to make our own IL-6 monoclonal antibodies and may do so in the future, we opted to purchase a pair of IL-6 antibodies suitable for use in an ELISA. Various IL-6 antibody pairs were tested until we identified a suitable pair that performed adequately. The pair was selected based on sensitivity, specificity, price, shelf life, and acceptable delivery times. So our PBMCs, together with the IL-6 ELISA and endotoxin standard, yield an LOD approximately 0.01 or below. So once the MAT had been validated, we then began the process of validating each product in a formal product-specific validation. Here we followed the guidelines in the MAT chapter 2.6.30 and considered the following aspects. Interference testing, in other words, does the product matrix interfere in any way with the MAT? This is done by spiking product dilutions with a known quantity of endotoxin and determining the percentage endotoxin recovery, with the recovery ideally being in the range of 50 to 200%. We also detect NMPs. Here, the product dilutions are spiked with a specific NMP to show that if NMPs were to be present in the product sample, they would be detected. And lastly, we consider interference in the detection system, where we consider whether the product will interfere with the detection system, i.e. the ELISA, in any way. Here, an IL-6 standard curve is spiked with the lowest product dilution, and you should really see very little difference between the two. After the product-specific validation is conducted, we then prepare a validation report and submit this to our local regulator for approval. All PDMPs produced at NBI are compatible with MAT using method 1, or previously known as method A, except for one, which requires method 2 or method C testing. So once the validations had been completed and regulatory approval had been granted from SAHPRA, we then began the process of implementation. So this covered two aspects really. Firstly, training of the QC analyst. So this was to be done on both method 1 and method 2. Our training took place over a period of 8 months and consisted of theoretical training on the mat, practical hands-on training to master the various techniques, and lastly, competency training, which involved inter-lab testing at NBI, where the same products were tested by both the QC analyst and a member of the MAC development team to ensure accuracy and reproducibility. A MAC testing laboratory had to be set up as the current QC or microbiology labs were not able to accommodate the additional equipment needed. We purchased new equipment and this was validated prior to implementation. So if I move on now to the last slide, really, this focuses on a number of challenges that cropped up along the way. So if we look at technical challenges, obviously I've already mentioned PBMC processing was our first big challenge. We needed skilled laboratory staff, and it was and still remains an extremely labor-intensive process. The MAT is a technically challenging assay. It requires excellent pipetting skills, and a good knowledge of cell culture and proper handling of cells is also advantageous. We did find method 1 more technically demanding. However, ironically, we've had more issues with the implementation of the method 2 product, and we are currently working to resolve this. If we look at the training of the QC analyst, like I said, this was over a period of 7 to 8 months. Ideally, more time is required. And it's essential that the analysts are not only capable of performing the MAT, but they have an excellent understanding of the assay and knowledge of why certain steps are performed the way that they are. This helps when troubleshooting or unexpected results occur. And then obviously, last, with your laboratory facilities and equipment, if you're going the route of processing your own PBMCs, you need to make sure that your centrifugation capacity is sufficient and that you have other necessary equipment, specifically rate-controlled freezing containers or a freezer, and that you have access to liquid nitrogen vapor phase storage. And then also with performing the MAT, we found obviously ergonomic pipettes to be essential. The analysts are doing a lot of pipetting for quite long periods of time, so good quality ergonomic pipette goes a long way. And similarly, buying good quality compatible tips for accuracy's sake. And like I mentioned before, we have one product that is not has not been validated with the MAT. It's proving to be particularly challenging and is showing significant signs of interference, but we are working towards resolving these issues. Yeah, and that brings me to the end. I would just like to thank the members of the NBI product development team for all their assistance and support during the development phase and the validations as well. And yeah, thank you to the HSI for affording this opportunity to NBI to present at the workshop. Thank you.
Thanks to you, Jacqueline. It has been very interesting to see the strategy and decision-making process and the challenges that you have been facing. We will probably see within the panel discussion about some of the issues that you also highlighted in terms of accessing to cost-effective kits, reagents, and also all the import challenges that you face. It's something that probably other countries are facing. So thank you very much for being so clear on that specific points. We will definitely get back to that in the panel discussion. Again, for the interest of time, I would move ahead to our last but not least presentation of this blood-derived products section. Thank you, Jacqueline. And I'm actually happy to give the floor to Ingo Spretzer from the Paul Ehrlich Institute. Ingo, I'm bringing up your presentation right now.
Yes, thank you.
Let me know when you can see it.
I'm still waiting.
Is it up?
That. Yeah.
Great. Thank you.
Thank you. So thank you for the organization of the workshop and for the invitation. So I'm going to talk about our experiences with our in-house MAT on blood-derived products Next. I have to do the disclaimer. So of course I'm a member of the Biological Institute, but this is clearly my own presentation, and especially my view on albumin is my personal view and not the current PI view, as you will see. Next. Very brief outlook. I will explain the situation we faced when we entered into that topic about the issue of blood products and their reported pyrogenicity, how the European directive affected pyrogen and endotoxin testing, the new pyrogen strategy in Europe, and the consequences, the putative consequences for drug import and export. So I have no fully clue on that, and the global future of pyrogen and endotoxin testing. Next. When I entered this topic with my PhD thesis, if you look at the available assays, the rabbit test in a way stood out because nobody really knows about the sensitivity of this assay. It depends on injection volume of the drug to be tested. There's no positive control, no standard curve, no spike and recovery. Maybe one of the reasons why people like to stick with the rabbit test, it's really complicated to do an invalid rabbit test because there are no controls. On the opposite, all the other assays, which we know, all the BET variations, including the recombinant ones and also the MAT, there you know the sensitivity, you confirm the sensitivity in every single assay, you have a standard curve, you do the spike and recovery, and so on. On the other hand, I agree, there will be more chances to have an invalid MAT than an invalid rabbit test because there are controls in the MAT and there are no controls in the rabbit test. Next. It's interesting if you look back that there have been really early reports about pyrogenicity of albumin, human serum albumin treatment. So from 1978 or from 1995. So there's something about especially albumin which always drew our attention. Next, please. So the pretty early MAT versions using whole blood or PBMC obviously are kind of well suited for blood products, which is not that surprising. I mean, if you're testing blood products in a whole blood environment, it's not really a different story for them. So there have been very early publications from around the 2000s that you can differentiate between endotoxin and non-endotoxin pyrogen in marketed drug batches. There have been comparative studies, and my colleague Rolando already mentioned the study done in Cuba on truly pyrogenic batches. So we really have the impression from the beginning that the MAT really has benefits here. Next, please. Interestingly, the MAT revealed also in 2 cases some very rare immunological issues between rabbit and human assays. There's a report from 2019 that a product which was produced from a plasma pool containing donations of 4,500 donors was failed in a rabbit test, and they didn't believe it, and it came out that 1 out of these 4,500 donors had special antibodies that makes every rabbit test reactive. But these batches then, as this was revealed, were released by the MAT, so the MAT saved this batch. And a similar case was reported in 2023 by Professor Turicek from Takeda, the Eau Claire incident, very similar to the case described by Servos. So the MAT, it might be better to stick in the target species. In our case, that's the humans. Next, please. So we did the rabbit test at the Paul Ehrlich Institute, and time by time we were challenging the rabbits with endotoxin just to get an idea about the sensitivity of our assay. And according to our studies, we concluded that the most sensitive rabbits in our case might start to react at the concentration of 5 endotoxin units of the WHO LPS, which would be 500 picogram per kilogram body weight, but only some animals, not 100% of them. As you can see here, it's published. Next, please. Now we concluded if this was our result. So the maximum injection volume according to European pharmacopoeia, depending on the product, is 10 milliliter per kilogram body weight of the rabbit. So if the 500 picogram LPS would be distributed within these 10 milliliters, this would make 50 picogram LPS per ml. So the most sensitive rabbits might pick up that. But if you compare that to the MAT and also to the BET, you have to be aware that's more kind of theory. These assays get more and more sensitive. Like in the BET, you might be able to detect endotoxin like 1 picogram or less. So of course, if you switch the assays, or if you have to switch the assays, you always have to think about what kind of sensitivity you have to achieve, and maybe not going to go too sensitive. Next, please. So as we did batch release of products doing the RPT, we took the chance and tested all of these batches, which we tested in the rabbit test in parallel, not at the same day, but close to that, in our monocyte activation test, which is an in-house assay based on cryopreserved human whole blood. So no isolation of the PyBMCs. And on purpose, we have chosen a relative insensitive MAT, still able to detect at least 50 picogram of RSE per ml. So it would resemble the most sensitive rabbit test, but we didn't go even further with the sensitivity. As we only received the good batches from companies to do the national batch release, it would be not helpful if we perform a much more sensitive assay, because then the batches will be stuck in our lab and we cannot release them. So it's a kind of a skewed validation because we only receive good batches, but we have a good correlation between pass and pass. But we also have access over time to 7, now it's more, batches which failed in our side, or where industry told us we have a pyrogenic batch, are you interested in that? And there we also have congruency, so it failed in the RPT, it failed in the MAT. This gives us the impression that our MAT has a good balance, being as safe as the rabbit test, but not being oversensitive, because we did not want to pick up or to exclude good batches from the market just because we are 100 times more sensitive than a rabbit test. The rabbit test seems to be safe. We just wanted to replace an animal test by an in vitro assay. Next slide, please. Now, if you look at commercial MAT setups, we also have seen in other presentation, some of them really have very low sensitivities, like picking up 2 picograms and so on. That's okay, you can do that. But most of the MATs which we know have a small dynamic range. So if you go to this low sensitivity, you really push the curve to the left, so your dynamic range will be really small. And I'm not sure if this is always necessary. Typically, if you have interference problems in the BET, you just buy a lysate with a higher sensitivity, but you stay with the same volume mix between sample and lysate, and then you can dilute more and typically overcome interference. Now, getting higher sensitivity in the MAT typically means you need more of your sample in the final reaction mix. Now, if you have an interference problem, increasing the amount of sample in your final mix won't help you with the interference problem. So here we really have a difference between how to overcome interference in the MAT or in the BET. And one problem of the MAT is indeed this very small dynamic range. Maybe comparable to the BET, you also have endpoint versions, which nobody is really using, whereas everybody's using kinetic versions because they have a much higher dynamic range. Next, please. Now I've explained to you how we kind of estimated and calculated the sensitivity of our RPT, but we really have to say that there are conflicting data. So Caroline has published here data where the lowest endotoxin concentration giving a reaction in the rabbit is 20 international units. So if 5 international units in this experiment would have raised a significant result, I'm not sure. And previously in 2023, there was a publication by Thurman et al., and they had issues in getting positive reactions in the rabbits with RSE going up to 30, 35 endotoxin units. So we have to be clear about that the estimation which everybody is using, that 5 endotoxin units per kilogram body weight will make every rabbit reacting might be pretty hard. I think this is the lower limit of detection. Many rabbits won't react to that. So if you base your calculations on that and then choose a much more sensitive MAT, you really challenge your product if you didn't improve your production quality. Next, please. So do I know of issues in testing blood products? Yes. Stankwin has published data on using either fetal calf serum or human serum albumin. Both have pros and cons. I know from some users which are in discussion with the PI because they want to move or have to move, that they have issues with spike recovery. Mostly it's below 50%, but above 200% is reported too. One case was interesting because the company finished the product specific validation, switching from the Rabbit to the MAT. Everything was fine. Then suddenly they found in frequent batches after implementation a spike recovery above 200%, and were in discussions with us because they say it's a problem of the MAT kit supplier. The cells have changed, and of course we were not fully convinced it might not have been the product. So fair enough. Some users have issues with the MAT because you have these quality controls like spike recovery, which you do not have in the Rabbit test. But if there are two methods, so if you want to compare your product against a standard curve, which includes spiking, you choose method 1. But if you have issues with that, you still have the option to go for method 2, which is a batch-to-batch comparison. You don't have these options in the Rabbit. So we already delivered 2 methods for you to choose. Next slide. Well, if you have spike recovery issues in method 1, you have 2 options. You can try to optimize your setup, or you can try to validate method 2, where no spiking is included. If the recovery is below 50%, you could try to increase the spike. You could try to use a less sensitive MAT, because as you spike in the middle of the standard curve, if you go for less sensitivity, the spike will become bigger. Less sample means less interference in the MAT. And even with a sensitivity of only 50 picogram LPS per milliliter, you outperform any thinkable RPT. And you might do sample pretreatment. The recovery above 200%, you could try with a smaller spike, but here I could think that maybe method 2 might be better. And of course, we also have to discuss if a spike recovery above 200%, is it that bad? Next slide. Well, if you see during product-specific validation switching from the Rabbit to the MAT or with a new product, that your product gives you a spike recovery above 200%, you have revealed an unknown characteristic of your drug. It enhances LPS-induced pro-inflammatory response. That's something you should know about your product. So you should maybe rethink the LPS limits for your product. So if all of your batches show that, go to method 2 or find another solution. If this happens with single batches, it is suspicious because then your drug production is not really consistent, which can be the case for biologics. So some people contact us and tell us they have PPC issues below 50% or above 200. They would like to stick with the RPT where no spiking is done, but I would say scientifically that's not a solution. The problem is still there. You still ignore it by not spiking. So we are still in discussions about that. Next, please. So the Rabbit tests will be removed from the European Pharmacopoeia in the 12th edition. So all of the texts, including the text for albumin, RPT and BET are not mentioned anymore, but there's a citation of this new chapter Pyrogenicity, which includes all of these now available assays. That makes MAT and RSC official and companion, at least if these assays are mentioned in the specific monographs. But those texts include these big chapters like substances for pharmaceutical use or parenteral preparations. So a lot of preparations are affected. And the Japanese Pharmacopoeia just released a draft for Monocyte Activation Test, and the USP is hopefully working on it. Next, please. So in Europe, the situation is relatively clear. But if you want to import to Europe, it might be a little easier for you to use alternative methods. If you want to export from Europe, it's a little more complicated because many other pharmacopoeias do not recognize currently these assays as official and compendial. But I think most of the regulators are open if you deliver sound and good data. So we have seen MAT long time ago being accepted in the US in a certain case, and hear good news from that. Japan is on the move. We see it in other countries. So the alternatives are really on the run. Next, please. So it's important, your choice. So we did not want to replace the testing for pyrogens. We wanted to replace the animal test by the MAT. If you choose to switch from the RPT to the BET, that's a reduction from pyrogen testing to endotoxin testing. For blood products, what we currently see is that most people try to switch from the RPT to the BET instead of the MAT, which I think is a pity. And in the case of albumin, me and Rolando and also Marike have the impression that the MAT is the method of choice to test albumin. The Limus assay makes no sense for albumin. Next slide, please. As Sanquin has shown with these 4 batches, so the BET was blind for the pyrogenic batches, which is pretty bad. Next slide. So I would combine the benefits of the BET, the MAT. I would do the BET during production. You have many samples, you do the quality. And if then finally you had a rabbit test and now replace it with another BET, you still know nothing about pyrogenicity. I would replace the RPT with the MAT, and then you have the full picture, including BET and MAT. Next slide. So the future of the BET, I think, is recombinant versions, and for the MATs, optimized MATs, including NEP standards, what we have heard about today, and engineered cells. Next slide. So what did we achieve? We replaced an animal test by a superior in vitro assay, which is predictive for humans. Thank you for your attention.
Thank you very much, Ingo, for your perspective and also showing the different strategy in case of some of the issues that you might encounter. Hopefully this is going to be helpful for our attendees to learn about the possibilities that they do exist. Again, I will encourage the attendees to drop their questions into the Q&A. And again, in the interest of time, so to not lose any time for our panel discussion afterwards, I would like actually to move to our next session that is dedicated to the vaccine. Before that, I'm really grateful for all the speakers of the blood-derived products session for their case studies and their interesting examples. So thank you very much to you all. And it's here with me for this vaccine session, Dr. Eliana Coccia from the Italian Institute of Health, and she's actually going to introduce the speakers for this session. Eliana, thank you very much for being with us, and I let you guide us through the session. Thank you.
So as you see from the program, now we have 4 talks dedicated to the experience that our colleagues had in testing some vaccine. 2 vaccine with no pyrogenic vaccine and other 2 with intrinsic pyrogenicity. So let's start with Dr. Ottavio Presgrave from Brackman Institute. Ottavio, are you here?
Yeah.
Okay,
yeah.
Nice to see you again.
Nice to see you again.
So, Laura,
Yeah, sure.
are you sharing or Ottavio is doing?
I will share.
Okay.
Yes, Ottavio is. I'm here in case something is not working.
That's good.
It's all good, Ottavio.
Okay.
You can go. Yeah,
Okay.
thank you.
Okay, thanks. I hope you can hear me clearly. Well, first of all, I'd like to thank the invitation for presenting this part of our work. I'd like to thank the organization of this event, especially to Laura for this invitation. Well, I'm going to talk about the use of MAT for yellow fever vaccine quality control. My name is Octavio Presgravia. I'm the coordinator of Brazilian Center for Validation of Alternative Methods. And well, this is an article we have published at ATLA in 2018. It's a study that joined some unities of Oswaldo Cruz Foundation, which is a big research center in Brazil and South America. And before starting, I'd like to make some remarks that Bill Manguinhos produced yellow fever vaccine, and both for this study we used both fresh and cryopreserved whole blood, and IL-6 showed a lower response when compared to IL-1 beta, and that this study was fully conducted by Catherine Matos, that is attending to this event and can help me to answer some questions if necessary. Okay. I just talk about the most important results of this study, some highlights of this study. Okay. Well, we start this study doing the recovery, studying the recovery of the LPS contamination yellow fever vaccine, and as you can see, most of the dilutions were between the range of 50 and 200% of recovery, and this is for IL-1 beta, and this other is for IL-6 marker result. The exception was the 1 to 160, that was higher than 200%, but we decided to use for both readouts, IL-1 beta and IL-6, the dilution from 1 to 10, okay, because in both cases it reaches the 100% of LPS recovered. So we decided to use this as our dilution. You can see here in the IL-6 the recovery of 100% again. Well, so we tested the ability of the MAT to detect different pyrogens when we spiked the yellow fever vaccine with LTA, the lipid teichoic acid from gram-positive bacteria, and we can see in the left graph the response of different concentrations of LPS, and the dark bar is the control, the sodium chloride spiked with the LPS concentration, and the gray bar, the yellow fever vaccine spiked with these concentrations of LPS, and the right graph the same, but spiked with LTA. So we can see that the response is very clear, and there's in case of LTA, no such difference was between them, the response of control or vaccine. It showed that the vaccine spiked with both LPS and LTA response to the MAT. And here is the correlation between the OD of LPS and OD of the sodium chloride and the yellow fever vaccine, both for LPS or LTA, and it showed a good correlation response. Okay. Here is just to highlight the LTA spiked yellow fever vaccine in different concentrations in order to show that there was no interference, and the response was very clear. We used this as a control, as a positive control, the 0.5 EU per ml of LPS, and the response of LTA was very good and very positive, both for IL 1B and IL-6. And here it's just a comparison between MAT and LAL, just to show that we evidently know that LTA is not detected by LAL, but it shows that MAT can detect it for the yellow fever vaccine spiked with LTL, and when LAL is used, it's not detectable. So it shows that MTA can detect non-endotoxin pyrogens in yellow fever vaccine. In conclusions, we showed that the yellow fever vaccine monograph recommends testing only for endotoxin pyrogens, and assays for the presence of other pyrogens are very important, and especially for the quality control for agencies and quality control laboratories, in order to exclude potential contaminations with non-endotoxin pyrogens. We investigate the ability of MAT to detect non-endotoxin pyrogens in LTA spiked vaccine, as well as we have done with LPS also, and so it was we found very good results in this, and showed that the contaminations with LPS and non-endotoxin pyrogens, especially LTA, is detectable by MAT. And our findings reinforced that the possible non-endotoxin pyrogen contaminants could be detected by MAT. And just for your information, I'd like to show that we have already tested MAT in any other cases, such as hyperimmune sera, especially and snake venom sera, and rabies, and scorpion and spider sera. Okay, we have some other articles about use of MAT in quality control. This is another article we published by using MAT for the quality control of meningococcal conjugate vaccines. Also, the same group of the 2 units of Oswald Foundation that we studied, and we showed that it was possible also for meningococcal vaccines to use MAT in this quality control. And these are some the reference, complete reference of some of our articles, including the use of MAT for air contamination detection and sera, hyperimmune sera products and some vaccines also. So once more, I'd like to thank the invitation for this event and to show a part of our studies using MAT. Thank you very much.
Thank you, Ottavio. We really appreciate your presentation. You were also on time, so perfect. I think since we are running late, I think that we'll move to the next speaker. And so it's Marilena Etna. She will talk about her experience on tick-borne encephalitis virus vaccine. And so, Marilena, you are here. Okay, so the floor is yours.
Okay. Can you hear me? Can you hear me?
Yes, yes.
Yes, okay, perfect. Okay, thank you, Eliana, for the introduction. Good afternoon, everyone. My name is Marilena Etna. I'm a researcher at the Department of Infectious Disease of the Italian National Institute of Health. And during my talk, I hope to address some of your questions that we received with the survey you completed. And I hope to address this question by describing our experience at the Italian National Institute of Health with the Monocyte Activation Test that has led to the foundation in our department of an MIT unit that from 2020 is involved in vaccine batch release. Our experience with MIT started a few years ago thanks to the participation to a project granted by the Innovative Medicine Initiative and called Back to Back. The goal of this project was to develop non-animal methods for the assessment of vaccine batch-to-batch consistency. And in this context, one of the aims of our unit was to adapt and optimize the Monocyte Activation Test as a replacement of the Rabbit Pyrogen Test for the pyrogenicity testing of tick-borne encephalitis virus vaccine. Let's start with the product. The tick-borne encephalitis vaccine is an inactivated vaccine adjuvanted with aluminum hydroxide. The active substance of the vaccine is the virus, inactivated by formaldehyde. The TBEV virus is a positive single-stranded RNA virus belonging to Flaviviridae family and possessing a small envelope with 3 structural proteins. Thus, by looking at this structure, there are no molecules able to intrinsically confer pyrogenicity to the drug product. However, if we go through the European Pharmacopoeia, the monograph foresee the test for pyrogen for this vaccine on the final lot. And the reason behind this necessity mainly rely on some critical steps in the production process, namely the embryo harvest from chicken eggs and the virus propagation that could expose to the risk of incorporation of bacterial, viral, or cellular contaminants in the final products. Indeed, in addition to endotoxins, as we know, many other molecules that we can broadly call non-endotoxin pyrogens can stimulate in human the fever cascade and all the worst reactions that are related with the response to pyrogens. And if we consider the non-animal methods that are currently available for endotoxin and pyrogen testing, we know that by recombinant factor C test, we can assess whether in a product there are endotoxin. But while MIT is suitable for the assessment of both endotoxin and non-endotoxin pyrogens. Thus, by considering that in the final lot of the TBEV vaccine can be potentially present both endotoxin and non-endotoxin pyrogens, we decide to apply and optimize the monocyte activation test for the assessment of the pyrogenicity level of this vaccine. And all the data I will show you in next slide are part of this paper that we published a few years ago in collaboration with some colleagues of the National Center for Control and Evaluation of Medicines of our institute, and also in collaboration with Paul Ehrlich Institute and GSK. Since diverse pyrogens could potentially be present in the final lot of TBEV vaccine, we decide to use cryopreserved peripheral blood mononuclear cells as a cell source. Indeed, primary cells possess a wider portfolio of cellular receptors respect to cell line, and thus they are able to sense many pyrogens. Thus, to qualify cryopreserved peripheral blood mononuclear mononuclear cells, we assessed cell viability and the reproducibility of the response of cryopreserved PBMC to scalar dose of the reference standard endotoxin over time. And by monitoring these parameters at 6, 12, and 18 months, we found that the viability of PBMC properly stored in a liquid nitrogen tank remains stable up to 18 months. And similarly, also the response to reference standard endotoxin in cells towed after 6, 12, and 18 months from freezing remain stable up to 18 months, thus indicating that these cells can be used for a long time. Then to choose the readout of our assay, we analyzed the release of the 3 cytokines that we can consider the endogenous pyrogen, since TNF, tumor necrosis factor alpha, interleukin-6, and interleukin-1 beta are the 3 cytokines responsible for the initiation of the fever reaction. And in this context, we evaluated the release of these 3 cytokines after stimulation of peripheral blood mononuclear cells with 2 doses of the reference standard endotoxin and 2 doses of 2 non-endotoxin pyrogens, namely the agonist of toll-like receptor 7/8 R848 and the synthetic agonist of toll-like receptor 2 FSL1. And as you can easily observe from the green bars of this graph, IL-6 was robustly produced by PBMC as compared to tumor necrosis factor alpha and interleukin-1 beta after treatment with all the 3 stimuli. Moving now to preparatory tests, we firstly assessed whether the experimental condition employed for the setup of our MIT ensure a linear response to different doses of the reference standard endotoxin. And for doing this, we performed some analysis with the 4 replicate of at least 4 endotoxin concentration that, as you can see, show a significant, statistically significant regression in the response to the different doses of the reference standard endotoxin, and a not significant deviation of this response from linearity, thus indicating that the assay is in agreement with pharmacopoeia requirement. Then to verify whether the vaccine interferes with the detection of endotoxin contaminants, we spiked into different vaccine ratio a fixed amount of the reference standard endotoxin, and we evaluated the recovery of this reference standard endotoxin. And we found that starting from vaccine dilution 1 to 12.5, the recovery of RSA was within the permitted range of 50 to 100%. We conducted a similar analysis also by using the excipient matrix, since if you remember at the beginning I said that this vaccine is formulated with aluminum hydroxide. And as you can see, we observed a similar trend, thus indicating that the minimal existing interference mediated by high doses of the vaccine in the final lot mainly depends on the excipient. And to verify whether the vaccine interfered at a technical level with the ELISA procedure, 5 doses of the TB vaccine were added to the IL-6 standard curve, and the interference was measured by considering the optical density value obtained by the IL-6 standard curve alone or combined with the vaccine at the 5 different dilution. And all the detected optical density value variation due to vaccine addition to the IL-6 standard curve, all these analyzed concentrations fall within the permitted range of plus minus 20%, thus indicating that this vaccine does not interfere with the ELISA procedure. And in accordance with the European Pharmacopoeia, the MIT optimized for the TB vaccine was also validated for its capacity to respond in a linear way to different doses of 2 non-endotoxin pyrogen, and also an assessment of the possibility of interference of the product with the detection of these NAPs was performed. And these tests showed that the MIT optimized for the TB vaccine show a linear response to different doses of the toll-like receptor 2 agonist, FSL1, and a linear response also to different doses of the toll-like receptor 7/8 R848, as you can probably see from this table, with significant regression of linearity and not significant deviation from non-linearity. And moreover, the vaccine and the excipient matrix were tested for the capacity to interfere with the detection of the non-endotoxin pyrogen R848. And as you can see, starting from, in particular, from dilution 1 to 100, we didn't obtain interference, thus indicating that both the vaccine and the excipient matrix does not interfere from starting from this dilution with non-endotoxin pyrogen detection. And in this slide, there is a summary of the main parameters characterizing the MIT optimized for the TB vaccine, namely the limit of detection and the assay sensitivity calculated for both the reference standard endotoxin and the 2 non-endotoxin pyrogens. But I would like to focus on the calculation of the contaminant limit concentration and maximum body dilution, since probably by our experience may be helpful for someone else dealing with pediatric vaccines, since this vaccine exists in both adult dose for adults and dose for pediatric dose. In the calculation for the limits for the pediatric vaccine, we considered as threshold of pyrogenic dose the threshold foreseen for parenteral administration, while as dose per kilogram of body mass, we considered the pediatric dose of 0.25 ml and the body mass of 5 kilograms, since it is a pediatric vaccine, and we calculated the contaminant limit concentration that resulted to be equal to 100 endotoxin unit per ml. Then we proceeded with the maximum body dilution calculation by applying the formula described in the European Pharmacopoeia, namely the ratio between the CLC and the limit of detection, and the maximum body dilution resulted to be equal to 2,700, meaning that we can use the vaccine in our assay until dilution 1 to 2,700. However, along our experience of optimization, we proposed also a new calculation for the maximum body dilution that consider the assay sensitivity instead of LOD, since the assay sensitivity is 1 point of the standard curve of the reference standard endotoxin or non-endotoxin standard curve, and is the lower point at the beginning of the linear part of the standard curve and not a calculated value. In this way, the maximum body dilution resulted to be equal to 1,000, meaning that the vaccine can be used in the assay until dilution 1 to 1,000. And based on the information collected until now, we established the final layout of the MIT applied for the pyrogenicity testing of the TB vaccine. We set up the modified version of this of the semi-quantitative method described in the European Pharmacopoeia by using cryopreserved human peripheral blood mononuclear cells as a cell source, and by using IL-6 release as a readout. In the plate layout, we included 3 doses of the vaccine alone or spiked with a fixed amount of reference standard endotoxin, and a 5-point dilution of the endotoxin standard curve, cell blank and ELISA blank, and also a positive control for the ELISA procedure, namely recombinant IL-6. And lastly, the assay was validated on peripheral blood mononuclear cell from 4 different donors by using 3 different TB vaccine lots. And according to ICH requirement, a precision study was also conducted to assess the repeatability and the intermediate precision of the method. This is my last slide. Thanks. I want to thank you all for the attention, and I'm happy to take your question, if any, or you can use my email address to send me directly your question. Thank you.
Thank you, Marilena. You already addressed some questions that were asked during the survey, so thank you for that. I don't know if you don't see any new question in the chat, so I think that we can proceed. And so let's move to the pyrogenic vaccine. And so I will ask Hilde Holdiek. Sorry, Hilde, I choose the easiest part of your family name to introduce you. Hilde is from NOMA, and she will give a talk on implementing MIT for a pyrogenic vaccine.
Thank you. I'm really happy to be able to be part of this workshop here today, and I'm looking forward to sharing the work that we have been doing. Like you said, I work at the Norwegian Medical Product Agency, and we are part of a European network of official medicines control laboratories, or OMCLs. And one of the things we do form OCOBR, which is batch release on vaccines. So that's what I'm going to be talking about today, is how we have implemented the Monocyte Activation Test in order to test a pyrogenic vaccine. So we've talked a lot about this already, pyrogens and what they are. They are substances that produce a rise in temperature in humans or in animals. And we have exogenous pyrogens that induce the innate immune system to produce and release endogenous pyrogens or cytokines, which again will stimulate the hypothalamus to produce prostaglandin E2 and cause fever. And it's because of this fever that we have pyrogen testing, and that's because of this that there's a requirement of testing parenterals with pyrogen testing. So that's what we are doing. We've already talked a lot about these two, the endotoxin pyrogens and the non-endotoxin pyrogens, the 2 different types. And as we all know, LPS is endotoxin, and that's a component of the outer cell membrane of gram-negative bacteria. And then we have non-endotoxin pyrogens that are peptidoglycans, bacterial lipoproteins, etc. The vaccine that we are working on is Bexsero. And Bexsero is a multi-component meningococcal B vaccine. And this is the first vaccine of its kind, and it contains 3 recombinant proteins and OMV. And all these components are adsorbed onto aluminum hydroxide. And this vaccine is a little unique in the sense that it's purified from the bacterial membrane, and therefore it's inherently pyrogenic, which means that the vaccine itself contains LPS and non-endotoxin pyrogens as well. So the vaccine itself contains fever-inducing components. So a little bit about the history of Bexsero testing at NOMA. The marketing authorization for Bexsero was granted back in 2013. And at that point, NOMA and NIBSC in the UK started performing OCOBR release of Bexsero. And at that point, it was endotoxin testing or pyrogenicity testing was performed by doing LAL and the Rabbit Pyrogen testing. And the Rabbit Pyrogen testing, as we know, detects both endotoxin and non-endotoxin pyrogens, so it's very appropriate for this type of vaccine. However, we saw that there was a high failure rate, both at the manufacturer and at the OMCLs when testing these vaccines in the Rabbit Pyrogen test. At the same time, we also use the LAL, which only detects endotoxin pyrogens. And we had a similar problem there, that there was very high variability in the results, and there was a high failure rate at the OMCLs. So due to this, it was kind of obvious that the RPT and the LAL was really not that great methods for testing this vaccine. So there was a big focus on implementing the Monocyte Activation Test in order to test this vaccine. And for several years, there was a very strong collaboration between NOMA, NIBSC, and the manufacturer to develop a method that could be implemented to replace the LAL and the RPT in testing this vaccine. So NIBSC, they succeeded a little earlier than the rest of us, and first in 2017, the manufacturer started using the Monocyte Activation Test to test this vaccine, and NOMA started using it at the same time. So this, you both, or all of you probably know, it's the European Pharmacopoeia Chapter 2630, which is the MAT. And it's two methods. There used to be three when we developed this method or implemented it. At that point, we used method C. Now this is method 2, which is a reference lot comparison test, where you compare the test vaccine with a validated reference vaccine. So that's what we were using. And as we've talked about before, there's a lot of different sources of human monocytes you can use and different readouts. And I will tell you a little bit more about what we did. So when we implemented the MAT, we isolated PBMCs from leukocyte bags, and we purchased these leukocyte bags from a local hospital, and we freeze the cells. And when we perform the MAT, we use these PBMCs from 4 different donors, and we stimulate these donors with a test vaccine and a reference vaccine. And this reference vaccine is a clinical vaccine that has been chosen and tested by the manufacturer. And then we dilute the vaccine, we do the stimulation overnight, perform an ELISA, and do a parallel line analysis. And I think some people in the pre-registration questionnaire wanted some more details. So I'm actually going to give you some details from the lab on how we perform this test. So first we get the leukocyte bags from the hospital, and we do a density gradient centrifugation. So you can see it here. We dilute the leukocytes, and then we layer them over histopac. We do a centrifugation step, and then you can see in the second picture, we get this layer of PBMCs and interface, and we discard the plasma, and we transfer this PBMC interface to a new tube, and we wash it with PBS. So after 2 washing steps, we resuspend the PBMCs in RPMI, and we count the cells. The last step then is to resuspend the cells in freezing medium, and we use DMSO and human serum for cryopreserving our cells. And we freeze them at a concentration of 24 million cells per vial. And we've done some stability testing, and we've found that our PBMCs are stable for up to 2 years in liquid nitrogen. So when we isolate our cells, we actually then have a lot of vials per donor that we can use for a couple of years for monocyte activation testing. So on the day when we actually performed the assay, we thaw the PBMCs, and we resuspend them in RPMI and count them and add them to the cell culture plate. At the same time, we dilute our vaccine and in a twofold serial dilution, both the test vaccine and the reference vaccine, and we add them to the cell culture plate, incubated overnight, and then the following day we perform a sandwich ELISA. And we actually coat our own plates with IL-6 monoclonal antibody. We add the supernatant from the cell culture plate, and then use biotin IL-6 antibody and streptavidin HRP. So you can see on the plates here, when you add then the TMB, you get the blue color, and finally the last step is when you stop the ELISA using hydrochloric acid, no, sulfuric acid. Yes, so this is then the next step is the data analysis. And this is just a picture of a hypothetical plate layout, and you can see the blue color, that's our reference vaccine. And we have that in 5 dilutions in quadruplicates. The red part of the plate is test vaccine 1, and the yellow is test vaccine 2, and the purple test vaccine 3. So with this setup, we can analyze 3 different batches of vaccine, and they're all compared to the reference vaccine. And so we read the plate at 450 nanometers, and then you can see here on the first graph, you have the vaccine dilutions starting at 1 to 1600 to 1 to 2500. And then you have increasing absorbance on the y-axis. So then you see the dose response curve of the reference vaccine and the other vaccine. So for this test, then we're looking at the dilutions, the 3 dilutions where you have the steepest slope. So we have 5 dilutions of each vaccine, and we pick the 3 dilutions where the slope is the steepest. And this point does not have to be the same in the reference vaccine and in the test vaccines. So for the reference vaccine, you can see that it's from 1 to 1600 to 1 to 6400, while for test vaccine 1, the red one, is from dilution 1 to 6400 to 1 to 25,000. So then you perform a parallel line analysis where you compare the reference vaccine to each test vaccine. And then that will give you a relative response. So that's what we're looking at with this method 2, is the relative response, or what we call relative pyrogen units of the test vaccine relative to the reference vaccine. So that's what you can see here. And in order to do this, we use CombiStats, which is a statistical program developed by the EDQM. So one of the very interesting things that we've found in working with this method, and that's been mentioned before, is the big donor-to-donor variability. So when we run an MAT, we always test 4 donors. So one vaccine is tested in 4 donors. And as you can see on this slide here, the response is very different from donor to donor. So when we isolate PBMCs, the first thing we do is to do a donor qualification assay. And there we run the reference batch in 10 dilutions, and we run a test batch in 10 dilutions. So for these 4 graphs here, it's the same reference batch and the same test batch run at the same dilutions. The only difference is there's 4 different donors. And since for this method, we are looking at the dilutions where you have the steepest slope, you can see that that's very different from donor to donor. So for us, it's crucial to do this donor qualification or assay first in order for us to know what dilution interval we can use for each donor. Because for this first donor here, we will use 1 dilution interval, while for the third donor, we will use a different one. So every time we then run our test, we will set it up at a different dilutions depending on the donor. Yes. Okay, so when we implemented this, like I said, we did it in collaboration with another OMCL and with the manufacturers. So these points are the validation criteria based on the ICH guideline. We didn't perform all of them. We did some, and I'm not going to go into detail on our validation, but I'll just show you parts of what we did. So one of the things we looked at was specificity, where we tested the IL-6 curve in the presence and the absence of test vaccine. And like it's been mentioned in other presentations, there shouldn't be more than a 20% difference. And we found that the test vaccine, in our case, did not interfere with the detection system. Another thing we looked at was accuracy. So the manufacturer had formulated a vaccine that contained 200% of each antigen while keeping the aluminum hydroxide concentration constant. So we used this vaccine and we did test it at a concentration of 200%, 100%, and 50% dilution. And the 100% dilution was used as a reference. And we ran this test in PBMCs from 20 different donors. And here you have the validity criteria, and our tests show that we passed them at all concentrations from 50% to 200%. And you can see also at the curve here that the geometric mean RPU that we found, the red line, is very close to the expected RPU that was calculated. For the linearity, our accuracy studies kind of confirmed, as you can see in this graph here, that there was no evidence of nonlinearity, so it was linear, and there was a significant regression as the sample concentration decreased. Also, when performing this reference comparison assay, linearity is a part of the assay validity criteria. So in every mat we test a reference batch and a test batch, and we choose the dilutions that give the steepest slope, and the assay validity criteria requires that for each range here, the p-value for nonlinearity must be non-significant. So before we, that's a requirement of the test itself. So these are just some of the things that we did in validating this assay. And the precision actually also, and for the precision, we tested 3 different vaccine batches in 21 donors over 3 different days. And we wanted to see how good it was at reproducing our results. And what we found, as you can see here, that there is a big variation in the RPU value, the relative pyrogen unit on the y-axis here, and each red dot represents a donor, and the line is the geometric mean of all the values from each donor for each separate batch. So you can see there's a big donor-to-donor variation, even when testing the same batch, as you can see with the variation here when we calculate the GCV. So we've also, in the labs we're collaborating with, they also see the same thing when testing this vaccine. So when we run the MAT, we do it as a 2-stage process where we look at the geometric mean. So what we have done is that we run every test in 4 donors, and we have a lower limit and a high limit and a retest region. So we run 4 donors, we calculate the geometric mean of the relative pyrogen units for these 4 donors. If it's below the lower limit, the vaccine will pass. If the geometric mean is above the high limit, the vaccine will fail. If it's between the low and the high in the retest region, we will test the vaccine in another 4 donors, 4 new donors. And here we have the specification. If it's below the specification, if the geometric mean of the 8 donors together is below the specification, it passes. If it's above the specification, it will fail. So after we had validated the assay, the next thing we did was to set a specification limit, as we found that the results can be a little different between the labs, we decided to set up our own specification. And this was calculated by our statisticians using a one-sided tolerance interval with a 99% population coverage and 95% confidence. And we tested then 32 different vaccine batches in 4 donors each, and that was the data that was used to calculate the specification in our lab. And that calculated a low limit, a high limit, and the specification. And that's what we're using when we are releasing this vaccine. If the batch does not pass the specification, we have an OOS investigation that we have to perform. And we do parallel testing with the manufacturer. So we're testing at the same time. And I don't think I'm going to go that detailed into this, but you might get slides later and you can look at it. This is how we have decided to do the out-of-spec investigation. And basically, we do test it in a lot more donors and several times. And of course, we also look to see if there's any laboratory reason for this. But the bottom line is that we do a geometric mean of 3 assays with all donors, either 8 donors or 4 donors, to see then if it's above or below the specification. So for my last slide, when we started with the Monocyte Activation Test back in 2013, we used the chapter 2630 and method C. Since then, there has been a whole new chapter that's now published in the European Pharmacopoeia, which is 2640, and that general chapter describes the use of the monocyte activation test to test vaccines containing inherently pyrogenic compounds, which Bexsero is an example of. And this chapter explains how to monitor the consistency of pyrogen levels when the pyrogens are a part of the vaccine. And in this chapter, you use an in-house reference lot, and you have to use this chapter in conjunction with the general chapter 2630. And this chapter was actually just implemented in July of this year, so we've been happy to see that. And that was my presentation. So if you have any questions, I'm happy to answer them at the end of the talk. Thank you.
Thank you, Hilde. You really gave us a lot of technical information that I think that are quite important for this type of webinar. So there is a question from Bernard Yulin. I hope I saw he is no, now is no, okay. Not no in the chat, so I think that probably we'll ask you some other information later in the roundtable. So I think that we can move to the next talk. Thank you, Hilde. We have Marieke Beck. She will, Marieke, are you here?
Yeah.
Oh, okay. So now Marieke will talk about the use of MIT to assess the reactogenicity of outer membrane vesicle vaccines. Okay, thank you, Marieke.
So I want also to talk about an OMV-based vaccines, but then in a development phase. So they are not registered yet. And this was a collaboration project together with Intrafak. So as just told by Hilde.
Marieke, can you?
Oh,
Marieke, can you put in presentation? Because it's.
Yeah, it's the wrong one. Yeah, I see it.
Okay.
Stop sharing. Yeah, I will. Save it. I think it's share. And now I'm, yes. If it's now going, I hope it's going okay. It's no, it's not.
Marike, if you go back, if you can change the display settings.
Yeah, I'm looking where.
Otherwise, I bring it up.
Display. I hope if it's now going. It's not.
Don't worry, Marike. I'm going to bring it up on your behalf.
Great. Yeah.
Okay, coming up soon. There it is.
Yeah. So. Yeah. Sorry for the. Just you can go to the next slide, Laura, please. So what are OMV-based vaccines? So they are outer membrane vesicles, OMVs, produced by gram-negative bacteria. And as you can see here, they have multiple pattern recognition motifs on there. So you have the LPS, as just indicated by Hilde, but also other molecules like peptidoglycans and other lipids available. The advantage of these vaccines is that they contain the complete patterns of reactogenic molecules on the membrane. However, they cannot grow and replicate and are therefore safe to be used. However, you don't want to have them at too high levels because then you can get side effects like fever, which are needed a bit, but not too high because then you have problems. The OMVs can be used as a vaccine against the bacteria from which it was derived, or it can be used as an adjuvant delivery vehicle to which peptides are linked, and then it will deliver also those linked peptides. This is also developed as a vaccination platform by Intravac, which is a company also based in the Netherlands. Next slide, please. So how do we want to measure, or how can you measure wanted reactogenicity when you are testing OMVs as a vaccine? Next slide, please. So you could use, of course, different pyrogen tests, which all have their advantage, disadvantage. So you have, for example, it could be a RPT, the Rabbit Pyrogen Test. However, then the procedure is usually adjusted to be able to test these samples because they already cause a fever. Samples are injected intramuscularly or they are diluted before injection. So it doesn't really reflect the correct formulation. The other option is the bacterial endotoxin test. For OMVs, this has the advantage or the disadvantage that it's only detecting endotoxin, while the OMVs will contain also non-endotoxin pyrogens, which are then not measured. And the MAT can overcome these two problems. And it, as indicated by Hilde just previously, it is accepted as a safety and consistency test for Bexero at their release. Next slide, please. So the aim of this data and study, which I will show, was to develop an MAT procedure to assess reactogenicity of OMV-based vaccine preparations, and then specifically a pertussis vaccine from Intravac, for which there was not a reference lot available because it was still in development. So that was one of the major problems. What will we then use as a reference lot? And then we decided to use Bexero as a reference lot because that has a proven safety profile and we could use that to at least have an idea on how much reactivity we would need or not need. And we then used method 2, which was then known as method C, but that was also indicated by Hilda. So if you look at our publication in scientific reports, there you will still see that it was then method C, because from July this year onwards, method C has been removed from the MAT chapters in the European Pharmacopoeia. We would use the cryopreserved pooled PBMCs and FBS and human serum as serum sources, because we were so human serum can contain all kinds of antibodies to which people are vaccinated via the national immunization programs. And pertussis is one which is quite long already in the national immunization program. So we didn't know if that would affect the MAT responses. So we choose to start with both serum sources. And we would use the IL-6 ELISA as a readout. So next slide, please. So at first, we just tested a lot of dilutions at 4-fold dilutions to see how would the pertussis OMV and Bexero behave, and FBS versus human serum effects. So as you can see, the red lines are FBS-based samples. So the most to the right line is Bexero with the FBS-based MAT, and the red line to the left is the pertussis OMV with FBS-based MAT, and the blue lines are the human serum-based MATs. So you can see that the human serum-based and FBS-based MATs were responding quite comparably. So that indicated at least that the antibodies against pertussis, which are present in human serum, probably didn't interfere or did interfere, but that the responsivity to NEPs is better with human serum, and maybe that caused the effect that it was comparable with each other. As you can see here, the pertussis OMV needed to be diluted 200 times fold more than Bexero to be in the linear range of the assay, so it was more reactogenic than Bexero. And at later stage, together with other data from my study, it was also that this will not become the human dose because it was too highly reactogenic. And yeah, in this case, we decided to continue with the human serum-based MAT, since it's comparable with sensitivity, but it also better reflecting the human immune system. So next slide, please. So then we started looking at the robustness of the assay with human serum, and we tested at several lots of human serum to see what would be the effect. If you can see here, the gray lines are Bexero, and the black lines, they are the pertussis OMVs, and then the 3 different lines for each sample, they represent the human serum lots. And as you can see, the pertussis together with human serum lot 1, that showed a significantly lower reactogenicity. That could be due by a difference in anti-pertussis antibodies. But for Bexero, we couldn't see differences between the different human serum lots. So therefore, we, together with Intravac, they looked at the antibody composition for pertussis in the human serum lot. So click, please. And there they showed that the different human serum batches which we used contain different patterns towards antibodies. We couldn't really pinpoint what was the cause, but this at least made us think, and we then decided to really take care when we switch human serum lots, that we really take care and compare, if not only pertussis will be shifted, because that's then the difficulty here. They are not the same, the reference and the tested samples. So if you only affect your tested sample with a new batch of human serum, but don't affect your reference lots, that's not the best way. So therefore, for development and setting up this, the reference lot can be something different than, or at least a bit comparable to the product. However, for release testing, you really want to have a reference lot which is the same material stuff, like and not a different OMV, which is from a different manufacturer. So next slide, please. And then we also tested several PBMC batches, and there we didn't find any differences between the different batches. And then next slide, please. And we also looked at different Bexero lots, and we, at least for these 3 different lots which we had, they were not that different within the different batches, so they were quite comparable. But also when we are switching to a new Bexero lot because the other one is expired, then we will also, investigate if it's not too different from the previous lot and what is then the difference, so that we can, correct for that. Next slide, please. So we then also investigated if the pertussis, Intravac also wanted to remove or to replace LPS with a mutated LPS which has a lower reactogenicity. So that mutation was the LPXA mutation, and that was expected to decrease reactogenicity. However, if you see in the upper graph, the blue line is wild type LPS from Bordetella, and we have the red one with squares. Which is the LPXA isolated from, the filled squares, which is the I'm just telling it wrongly, sorry. So the 2 left ones, which are the most left, which have a nice S shape, they are the 2 OMVs, one with the LPS wild type and one with the LPXA mutation. And as you can see, at least in MAT, they are still quite comparable to each other. So the LPXA itself did not appear to do anything, at least in this case, when it's together with all the other outer membrane proteins. When we tested the LPXA isolated from Bordetella and the LPS and the normal wild type LPS from Bordetella, then we could see differences. So the LPXA mutation itself, only LPS, that is reducing reactogenicity. However, when it's in an OMV surrounding, then that reduction was not seen. So then we investigated, are there differences then in expression of other non-endotoxin pyrogens by reducing the LPS expression on the outer membrane? And that appeared to be the case. So when the LPS modification is present, then the TLR expression of the endotoxin expression on the outside of the outer membrane is somewhat disturbed. And during growth of the bacteria, it appears that then there is more TLR2 reactogenicity. So that is the lower graph. There you can see the responsivity of the cell lines to the different wild type OMV LPS and the LPXA. So you can see that the red line, which is the mutation, has higher TLR2 activity than the wild type. So probably due to the increase in TLR2 agonists on the membrane, thereby we don't see a difference between the LPXA in an outer membrane vesicle or the wild type LPS. So not always as you would expect. But yeah, that's science. So next slide, please. And in the end, we also tested if this setup with a reference lot of PEC0 could also be, and the MAT could be used to test different OMV, different OMVs which are derived from different bacterial species. And that appeared to be the case. So we could see that pertussis, which is the blue line, was still at the same concentration. So it was a high reactogenicity. The black one is the PEC0, and the Neisseria meningitis B is the purple one, which is somewhat lower than PEC0. And there was a Neisseria gonorrhoeae, which seem to be comparable in reactogenicity. So it seems that you can also use this setup for different bacterial species than only the ones which we tested more extensively. And then we compare, in the end, there was a nice comparison between MAT and modified rabbit pyrogen data. So it was modified RPT, which monitors for 30 hours after an intramuscular injection. And on the left side, you can see a lot of lines of the different samples which we tested, because we tested 100 microgram samples which were used during the setting up of the assay, but also 20 microgram samples, because it was appreciated that for the human dose it should be lowered. So therefore we tested those different concentrations, and we compared it to a wild-type pertussis vaccine, so a really vaccine which is used mainly in third world countries. And we could see that there was a nice correlation with MAT results and RPT results. And if you click one more time, I think there are additional blue, and could you click once more, Laura? Yeah, so there you can see that, one back. So there were appearing some blue lines and red lines to show that when you compare wild-type LPS OMVs with 100 and the 20 micrograms, that you can see a nice decrease in MAT responses and a nice decrease in rabbit pyrogen temperatures. And with pink line, you can see that for the LPXA mutation, you can also see that trend. So this shows that at least the temperature rise in rabbits is correlated together with the MAT results, which we found. So next slide. And these are then just the short conclusions, like we could use the MAT assay to determine reactogenicity during the vaccine development and also test different OMV-based preparations. You can use an alternative lot during development of the product, and but you should then switch to a true reference lot of the same material where you are going to test, of course, for release testing. The MAT for OMVs can be based on human serum instead of FBS, as human serum allows for more sensitive detection of non-endotoxin pyrogens. However, in some cases, maybe you would need FBS if there are interfering antibodies in the human serum. But at least for pertussis, that didn't appear to be the case. And we found a good correlation between MAT and a modified RPT. And then the next slide is a thank you to all my colleagues again. So
Eliana, you are on mute.
Okay, I was thanking Marike for this very interesting presentation, full of details that I think that all the attendees will appreciate. I hope so. So we are, I guess, perfect on time, probably 2 minutes in advance. So that's good, I guess. So I would really thank all the speakers for their presentation. And so I'll give the floor to Laura.
Thank you, Eliana, and thank you to all the speakers. I would like to invite all the speakers to come on camera because we would like to start right away our panel discussion. So hope to see you all here. Thank you very much for hanging on with us during this long workshop. It's very appreciated. We would also like to thank you again for sharing in advance your questions. We hope that through the presentation you were able to find some of the answers, but I think that it will be important maybe to still highlight some of the points and the questions that we received. But before doing that, I actually would like to thank Paul Stickings, that is also joining us for this panel discussion, and also from MHRA, and also Tong Wu from Health Canada that are here with us. And it will be very nice to have this conversation all together. Again, I'm going to bring up some slides with the questions that some of the questions that we received, and we selected really to hope to have some conversation between the different speakers. But of course, we hope to have also more feedback through the Q&A or the chat together with the attendees and the participants. So if it's okay with you, I would probably like to go back again to a very basic points, because in the transition from the Rabbit Pyrogenicity Test to the MIT, there is probably still some request or some needs for clarity in terms of the relevance to perform a comparison between the MIT and the Rabbit Pyrogenicity Test. And at the same time, some of our attendees, they are actually asking for some recommendation on how to set up acceptance criteria for new products. We saw some of the work that you have done, but I would like actually to check if you would like to comment a little bit on this topic. That would be nice. Please, Marlena, thank you.
Okay, I think this topics was well covered by the presentation of Ingo, of the today presentation of Ingo. What I want to add is that although we know that RPT has been historically recognized as the gold standard for pyrogen testing, in my opinion, it should be considered that we are dealing with medicinal products intended for human use. And although the rabbit immune system, this is an opinion from an immunologist, okay? And although the rabbit immune system is very similar to the human one, it's not identical, and some differences exist. And as we clearly heard today, in particular for non-endotoxin pyrogen testing, and particular for probably viral pumps. And so I'm not convinced that a strict comparison is mandatory, although if any available parallel data for RPT that will contribute to an assurance that RPT replacement with MIT is appropriate, may help, is welcome for sure. But I don't think it's a mandatory aspect in the transition. And moreover, as we heard today, we had some examples, experience. For example, the RPT, when was applied for the MMB vaccine,
ensured safe vaccine, but it was highly variable and prone to false positive results. And that is sometimes also the Rabbit Pyrogen test is wrong. The MIT, in the case of MenB vaccine, for example, was able to detect this vaccine batch as safe and allow the approval of this vaccine batch for marketing authorization. This is my personal opinion on this aspect on the comparison.
Thank you, Elena. Thank you. Octavio, please.
Well, I think it's a natural way to use RPT as a golden standard for a comparison with the MAT. But personally, I think that it's not necessary anymore. At least since the Rosenstein article that changed the variation of temperature in the United States pharmacopoeia, I think it's very clearly established that the response of human beings of a limited dose of LPS. So I think that we can use this data, include because the MAT substrate is human blood. So I think we can use as the limit or as comparison the lower dose that cause fever in human of LPS or in terms of units of endotoxin. So I think this is more comparable than RPT.
Thank you, Octavio. Please, Caroline.
Hi. So I think something that's really important to consider as well is how you're using this test. And in the case of the MenB vaccine of Bexsero, we knew it was pyrogenic and we were trying to use it to quantify the amount of pyrogen there, whereas the Rabbit Pyrogen test is designed to measure contaminants. So you've got a qualitative test and a quantitative test. So it can be very difficult to actually compare those two when you've got this quite different readout, which was one of the big hurdles we had. And as we've said, we had many failures in the Rabbit Pyrogen test. And if you go back, I know Ingo showed that very important figure where you see the amount of endotoxin, which starts to generate this temperature in rabbits. And what we could see is that we were working very much on the borderline. So we had to dilute the vaccine hugely before we actually gave it to the rabbits. So I think in the case of vaccines, which are inherently pyrogenic, it's a really difficult thing to actually do. I think for looking at blood products, it's slightly different, but still difficult. But back to the points that have been made by both the other presenters here is we have built up quite a big evidence base now of how you can, of how this has already been done. And it would be very good if people could use what's already there to show that, we do know we are measuring something which is linked to a pyrogenic effect, even if it's very difficult to say exactly if you have this readout in a monocyte activation test, you're going to get this temperature in a rabbit.
Thank you, Caroline. Please, Tonga, jump in.
Actually, I just want to mention that I'm in agreement with all these comments made. And one of the things is about MAT is the importance is to ensure that the commercial lots that they don't contain, they contain the pyrogen very, very similar to clinical lots that are shown to have good safety profile in clinical study. I think one of the important part is to set the right specification. And again, for vaccine products such as Bexsero have a high pyrogen content, and the PRT was baked, the test is using a dilution, and they should come with similar conclusion. I think if you're looking back at the presentation of colleagues Ingo presented, there are a concordance between the two assays in terms of the loss testing results. Of course, that is what is the specification set for MET assay. I think that is something we need to consider when we develop vaccines. Over.
Thank you, Tom. Any other comments on this? Okay, good. So let me see, there is something in the chat. Okay. Okay, thank you, Bianca. So, okay, so we will actually get to this question starting from this one, honestly, because we found this very interesting. Again, in terms of perspective about the perspective and strategy on the pyrogenicity testing strategy. So the question here is that will the endotoxin test be replaced by the MIT? So if an organization implements the recombinant factor C, it's still required that MIT has to be performed. So I hope that this could, your answer will be guiding the attendees to understand exactly when and how the different pyrogenicity strategy, being the MIT or the bacterial endotoxin test via the recombinant factor C, could intervene into their batch release strategy. I don't know who wants to get this question, and then we will add to the presentation in the Q&A. Please, Marike, thank you. Oh, Marike, you want to? Oh, okay.
Yeah,
Marike.
I want. Yeah, sorry. Yeah, so I think you still need MAT if there's a chance that you have non-endotoxin pyrogens in your products. And at some stages, you don't know if you're having non-endotoxin pyrogens or only endotoxins. So I think one of the approaches which are taken by clients of us is that they will only do MAT on the first 3 PPQ batches, compared with endotoxin testing, and then state risk-based assessments like we don't see non-endotoxin pyrogens. For batch release, we will only do endotoxin. We also, together with Ruud Daniels, have published a paper on MAT for monoclonal antibodies, so I can put that in the Q&A or comment session later on. Yeah, so I think MAT is still, and also for albumins, of course, I think you would need it at that stage when you know that there are other contaminants than endotoxin.
Thank you very much, Marike. And yes, please, if you can put the publication in the chat, it will be fantastic. Please, Octavio, and then Ingo.
Well, I'm a little bit suspect to answer this question because I'm always starting a MAT fan club, because MAT is one methodology I really believe, and so I think that the MAT will replace endotoxin tests, but I understand personally. I think that because of the LAL or RFC are more rapid to be performed. So I think that during a process of for an industry, maybe it should be recommended to be used in the process in order to see at least if there's no endotoxin. But as a final methodology, for a final safety result, I think that MAT must be used instead of any other endotoxin test, being LAL or RFC. I think that RFC is better than LAL because it detects all endotoxin and does not suffer any influence of glucan. But so I think RFC is better than LAL, classic LNL. And I think that for the final result, for the final product, I don't see anything better than MAT.
Thank you, Octavio. Please, Ingo.
Well, I hope that in the long run, the MAT will replace those endotoxin tests where pyrogen test should be done. So the rabbit test has been replaced in most cases by the endotoxin test, but some of these decisions from today's view are debatable, as we have discussed for albumin. The new European pyrogenicity strategy gives the option to the user to choose. If he has a test for pyrogenicity, he can do the endotoxin testing if he is doing a risk assessment and also doing practical data. And then there's a sound basis having practical data to do the BET. And as Octavio also mentioned, because of the timing, so BET takes 1 hour and classical MATs take 2 days, there is for sure in process, there are places for the BET, but for the final product release, as you have to guarantee that your product is not going to be pyrogenic, you cannot really do that with the BET. So I would like to see the mix of the BET where it is used and useful, and on final product, the MAT. And on the final product testing, an assay which takes 2 days or 1.5 days isn't an issue. Sterility testing will take you 7 to 14 days.
Thank you, Ingo. Please, Tong.
Actually, I just want to comment in terms of vaccine product. I think now the quality control of vaccines is not just about the final testing, and it's also rely on the process validation. And if we're looking at European Pharmacopoeia as well as WHO TRS, and there are many recommendations for both the control testing throughout the manufacture process as well as for the final product. And the endotoxin test has been recommended for many vaccine products because well characterized the product as well as the consistent manufacturing process. I think in that sense, when we thinking of replacing endotoxin assay or pyrogenicity, rapid pyrogenicity assay, we need to thinking of the assay performance itself. And as we just looking through the presentation, there are still some control about the mass assay, the use of cell line, the variability about PBMCs. And also, I think we have not discussed about when you're using vaccine reference as to calculate relative potency, the reference replacement also bring additional challenge in terms of assay control through a very long period life cycle management approach. So taking all of this together, I think replace endotoxin assay with a BET or recombitancy assay may still have a place. I think we still need to continue to discuss taking into consideration of the assay performance. Over.
Thank you, Tong. Yes, indeed. I guess that the more we are going to gain experience about the use of the MIT and how the recombinant factors is also going to place a role in the future, we probably might see a little bit how a combination strategy could really become an effective one, and what is going to be the future of the MIT implementation at all. So thank you. I would like to check if there are any other comments from the panelists on this topic. Otherwise, I think that we can get a little bit more in details about something that has been covered in some of the presentation as the challenges with regards to the commercially available kits. Because discussing with many stakeholders, especially from the low and middle income countries, getting access to kits, reagents might be a challenge. So I think that this question really represents a little bit about some of the challenges that are actually in place currently to the stakeholders that are actually considering right now the switch from the Rabbit Pyrogenicity Test to the Monocyte Activation Test. We have seen the case from South Africa and the pathway that follow, but I would like to actually get additional comments from your perspective, because our participants are mainly from the other regions of the world that are still experiencing some of those challenges. So please, Ingo, thank you.
The kits are expensive indeed. I mean, there's a lot of quality control in it, but what really drives the price, and the kit suppliers have nothing from that, is the transport cost. So if you ship on dry ice by plane kits from Europe to another place in the world, the shipping costs might exceed the kit costs. So I'm wondering if in those cases it wouldn't be better to have a local supply with cells of high quality, and I think that should be possible. Wherever blood for blood transfusions is processed, the leukocytes, the white blood cells, are filtered away. So typically they are discarded. These are the Buffy codes. These cells are of highest quality. I mean, the donors are selected. They are tested for infection markers. So the Buffy codes will be there as a waste in actually every blood bank. So maybe it would be good to get into the discussion with the International Society for Blood Transfusion. They cannot simply sell these Buffy codes for commercial use. They have to check the donor specifications. The donors have to know that, and they have to agree on that their Buffys, which typically would be discarded, would be used for other commercial use. But the cells actually are there in every country where blood transfusions are processed, and that could help. And of course, it's kind of more efficient than working. I mean, amounts of monocytes is simply bigger than if you do a small scale donation. So typically you are not going to go to your co-workers and take away half a liter of blood from them. They won't like it, but in a blood bank they can do it. So that would be for me one possibility, maybe to reduce the transport costs.
Thank you, Ingo. Eliana and then Octavio. Eliana, you're on mute.
Sorry, this is just my personal opinion, but I realized that the number of the supplier are increasing. So, and I guess due to this increasing demand for MAT, due to the new pharmacopoeia request, I think that the price probably in the near future could drop. So I hope that this will be the case. So I also would like to remind that it's also important to take into consideration that MIT, as we have heard today, is not a problem, only economical problem, but provide us a more reliable and robust way to test products that will be for human use. And so I think that we should keep in mind this also this feature of MAT.
Thank you, Eliana. Indeed, suppliers are increasing, but I want to go to Octavio, please.
Of course, I'm not the most appropriate person to talk about market or commercial, but first of all, I agree with Eliana that I think that as well as the MAT will be more used, I think that more suppliers will appear in the market. There's another way that that is possible is to use an in-house ELISA, but it's necessary to certify the quality of this in-house every time we make the plate. But there's a phrase here that one kit supports testing of only 2 samples. I think that it can be also suppressed if I think that only to samples if we perform all the dilutions that are described in the monograph, in pharmacopoeia monograph. I think that I believe it's a personal opinion. I believe that since we know the product, if we have already established the MVD and the LOD, everything in the product. For example, a vaccine, it doesn't change. So I think it's maybe it's not necessary to perform all the dilutions, all of that recommended of the lambdas and everything like this at the pharmacopoeia. So I think that since we know exactly the performance of the product, we can use more than one sample per microplate. So I think it's just a question of time, of knowing the methodology more properly. I think that soon it will be overpressed, suppressed, and I think that costs will be lower, and it will be possible to make more than 2 samples per microplate. It's my opinion. Thanks.
Thank you, Tavi. Please, Marlena.
If may add only comment, I think that from what I heard in this year from people from different part of the world trying to start with this experience with the MIT, probably the major issue is not the validation of the ELISA procedure inside the laboratory, while the major issue is the availability of the cells, in particular if they decide to use primary cell source, since there are some legal issues that are, in Europe we can manage this issue, and we have a clear legislation on this, but probably in other parts of the world there are legal and ethical issues from related to the use of blood and blood-derived products, and probably the major problem to solve in order to get to optimize an MIT at lower cost is to solve this problem of the cells. This is my opinion.
Thank you, Marlena, and we will definitely get on that point because we have an interesting experience to share from Brazil. But please, Rolando, jump in.
Okay. I am completely agree with the comments before. I just want to share our own opinion, no? Being from Cuba, a developing country, how we have solved partially this situation. I will encourage the using of cryopreserved blood, whole blood, pulled from several donors, have worked okay so far for us. And in addition, as Octavio said, the using of in-house ELISA. So we bought a matches antibody, and we titrate the antibodies, and once we get the title and with the background required for the ELISA, then we can conduct tons of ELISA using only one pair of matches antibodies. So the combination of in-house ELISAs, buying commercial matched antibody from thermal lab system, and the using of cryopreserved human whole blood cells as a source of monocytes have worked as the cheaper version in our case. We have tried several approaches, and this one seems to be the best one. You should need, in addition, reference standard endotoxin, international interleukin standards, but with this in your box, I think it could be the best way. Anyway, I should emphasize that this is still expensive, okay? It's still expensive. It's a challenging topic, and we have been concerned about this since years ago, trying to find out the best solution. It's true that one kit it costs around €1,000, and you can do nearly nothing. In addition, in the implementation stage, you will spend a lot of regions without any actual verifiable results, you know. So it's really expensive, but we have found this a kind of cost-effective approach to this situation like this. Okay, thank you.
Oh, thanks to you, Rolando. Thanks to you. It's definitely a very interesting experience yours, being in a very special situation. Please, Bianca, and then Paul.
Thank you, Laura. I just want to highlight something from the Brazilian perspective regarding this, and as part of the initiative led by Laura Viviani on the implementation of the Monocyte Activation Test in Brazil, we have a working group with the industry also. And as we have this normative resolution deadline finishing next month, that is this mandatory replacement of the Rabbit Pyrogenicity Test by the Monocyte Activation Test. And there were some concerns from the industry regarding relying on the direct donor of blood because this may not be feasible for the industry because of the high demand. But there were some concerns on using blood from the blood banks because we have specific legislation on the use of blood from donors. So we got in contact with the Anvisa coordination, the blood coordination, and we are working on a technical note just to make it clear that the remainings of the blood banks can be used by the industry to perform the monocyte activation test. So this is on the, we are managing to solve this issue now, and we hope that soon this will be clear for the Brazilian industry that wants to use the blood from blood banks.
Thank you, Bianca. Thank you. Yeah, this is a good approach that's we are trying to have where there is a specific legislation that it seems that is not allowing the use of donated blood, but again, talking with stakeholders is allowing us to actually find a solution. Please, Paul, jump in. Thank you.
Thanks, Laura. I wondered if there are no more comments from the panel to this topic, there are a couple of participants that have asked questions in the chat that we could maybe pick up. The first is asking the panel for any insights or perspectives on the relative pros and cons of using an engineered cell line with a, for example, a luminescence readout compared to blood or PBMCs measuring secreted cytokines. I can't remember who covered that in their presentation, but maybe they could offer their view on that. Thank you.
Thank you, Paul. Please, Marieke, please.
Yeah, so at this stage, we are still investigating the cell line. So with the luciferase, at least, so we have, but one of the drawbacks is maybe can be for us, at least with the vaccine testing, that it's only one genetic background. For batch release testing, as indicated by Ingo and others, that, yeah, it's less of an issue. But we are still investigating if the timing with those luciferase assays is okay or not. So yeah, so maybe it's too short for certain NEPs, but we don't know for sure. So we are investigating if that's the case or if there's something else happening with that. But they are quite new on the market. So with the luciferase, we don't have a lot of experience at this stage, but it's being continued.
Thank you, Marieke. Please, Ingo.
Yes, we have some experience in that. So typically the monocytic cell lines, they are derived from tumor patients. So they have damage which make them, they cannot die anymore, and you do not know the whole damage. So you can do that probably better using like iPSC-derived monocytes, because there you know what you have changed. And if you do that correctly, you will always not only have access to the same donor, but to the same donation. The reporting systems like luciferase might have the promise, and we see first results on that. You have much less pipetting, and this results in much lower CVs. And this is amazing, because for the classical MATs, we work with quadruplicates, which causes two samples per plate. But if you have CVs below 10%, like in the BET, we do not talk about quadruplicates anymore. We might talk about duplicates, and this will bring the pricing of the MAT much closer to the BET. So I see benefits for all kind of these cells. So sometimes you want to use the primary, why not? So they are closest to reality. And in some other cases, you want to have something which is really specifically reporting to that what you are interested in. So it's really, in the end, the user has to decide what is the best option for him.
Thank you, Ingo. Please, Caroline.
Hi, so the format that we use here is to use cells from a blood bank, just to sort of tell people. The question, what makes most reproducible results? Well, the difficulty there is at what stage you might want to be doing the testing as well. So not only have we've talked about as you go through the different manufacturing, but where are you with your product? Are you in the early stages? Do you know what it is that you're trying to measure? Because you want a reproducible method to measure the batch consistency, but often the issue we have is we're not entirely sure what is causing these pyrogenic responses. So therefore we want the full spectrum of the TLR receptors to be there, and we've seen issues with the Monomax 6 line where they weren't as responsive, and we don't exactly know why that was. We haven't been able to do it. So you may want to try and capture as much as you can of that donor to donor variability. I think I've been in many conversations about this, and I do see Peter Brugger's got his hand up, so that would be interesting. But catching variability of a population with 4 donors isn't great either. So that there are lots of difficulties here, but I do agree that if you know what you're actually looking for, and you know what you want to measure, there may be, moving down the line, some clever ways that we can engineer cells so that we know, and we can use things like the luciferescence. But I think you need to kind of take it one step at a time. And reproducibility, we hope we can bring that in by things like standardization, but you may not be able to get reproducible results in the first instances when you're using this test, because you're not quite sure what it is you're measuring and what the different donors might look like.
Thank you, Caroline. Thank you. Any further comments on this? So there is definitely the need to see more experience on this in this type of cell lines and see their performance. If there are no questions,
I think Laura, Peter has his hand up as well.
Sorry, I didn't see it. Sorry, please, yeah. Peter, can you speak? Okay, so just a second. We try to unmute Peter.
Just a second, Laura, I'll do it now.
Thank you.
Peter, can you please try to unmute yourself?
It was just a little bit problematic. Thank you very much.
You're welcome.
Thank you for having the chance to speak here shortly. I would like to speak out a mild warning about already going now to the next step with engineered cells and so on. I personally still are working on the exception of the monocyte activation test in US, and I think if we are doing it too much, too less overseeable, then this will be even more complicated for the American colleagues to accept this test. And we are preparing a study now showing what are the differences between testing single donors in comparison to pooled donors. And I think that for presenting this to the American authorities, we need at the moment the best monocyte activation test which we have. And this is my concern, that we have, first of all, we should see that the test, the MUT, comes into the USP, and also we would need a little change in the CFR. You know, in the CFR, it still stands for biologicals that you have to perform the rabbit test on at least 3 batches. And what we are really striving to do is that the American authorities, and I think these are not first line, the biologicals, the biologics, the biologists, and so on, but more people from the Senate or whatever, from the politician part. So I would encourage all of you. I have seen you have beautiful presentations here, and the test is going on and becomes more and more professional. It's absolutely in my sense what is done here. But I would like to ask you if you could help and support me in getting acceptance in the US. So that's my statement.
Thank you, Peter.
Thank you.
Indeed, we would like to make sure that there is as much as regulatory harmonization as possible, especially because we are seeing a lot of countries moving ahead in terms of creating chapters about the monocyte activation test. And the USA is definitely an important market, is an important regulatory agency. So for us, making sure that all the available data experiences that we share today, and it has been shared so far, is going to hopefully inform a transformation also on the other side of the Atlantic, at least from our European perspective. So it would be nice if you can share while you are keep working on your study about the comparability between single donor to the pool donors. It would be nice once you have a little bit more results, we can definitely discuss and bring up the topic again with these multi-stakeholder groups, and we can definitely further discuss about how to progress in the countries that are yet to be taken an official stand on this. So thank you for your comments, very appreciated. So we are at time, but I would like to actually get to one final question that, again, it's about regulatory acceptance and the potential complexity that manufacturers versus national control laboratories might experience when they decided to use different type of platforms. And this is something that, again, I would like the panelists to jump in and to make their comments, because since we are having a lot of the attendees in their process to develop their own assays, to have their conversation with their national regulatory agency and national control laboratories, is going to be very important to explain if you have some recommendation on how you have been actually experiencing this specific case study. So please, Paul, thank you.
Thanks. Yeah, a nice easy one to end the webinar with. This, of course, is not an issue that is exclusive to MAT. It's something that can occur with many different tests, and it's never a simple solution. But one point that I would make is that if there are available, stable, standardized reagents, such as the ones that Caroline talked about at the beginning of the webinar, you can at least answer the question as to whether the assay is performing consistently or not. And that can often be the first thing that you need to understand before you then further investigate the root cause of any discrepancy. So having those type of materials available is really important to help at least start the investigation into these type of issues.
Thank you, Paul. Absolutely. Any other comments from the panelists on this point?
You're muted,
You're muted,
Laura.
Oh, sorry. Sorry. If you want to jump in, but my perspective is, first of all, to encourage dialogue between the manufacturers and their own authorities. Just start early dialogue when you are developing your assay, while you are doing your product-specific validation, making sure that there is the possibility to share data and understand exactly what are the expectations from your regulatory agency, and also with the people from the national control laboratories that are actually going to do the test, because that is going to help you to solve even during the batch release testing phase, in case there is actually something that is not working, and avoiding to maybe lose batches of time and resources if something is not correctly or is not performing as it's supposed to be. Please, Caroline.
I agree very much with both points that have been made, and just to say again, our sort of what Hild presented showed where we first started off and we hadn't had those good conversations, and we ended up with the Rabbit Pyrogen test, but it wasn't just a straightforward, it was a modified Rabbit Pyrogen test, which we weren't ready for in the OMCL laboratories. When we then worked together with the other OMCL and the manufacturer, you know, it was a really good way to solve a problem. I know that not everybody will have that opportunity, but I can't agree more that a conversation between yourselves and the manufacturers, the NCLs and the manufacturers, is hugely valuable.
Thank you, Caroline. Any other comments? Some closing remarks or highlights that you want to give before I close this workshop? Okay, so thank you. I know that we were not able to cover all the interesting questions that you had, but we will make sure in the workshop report to address them all. So thank you for being with us, for your active participation. I would like to thank the scientific committee, of course, and the speakers for the great work and clarity that they offered and their experience. As the Animal Free Safety Assessment Collaboration, we are actually organizing other meetings on other topics. This is part of the project that we are running. So keep monitor our websites and the LinkedIn platform for the upcoming events. We have something dedicated to the veterinary piece. We will discuss about tetanus, diphtheria, and pertussis in an upcoming workshop next year, and also about the use of the high-throughput sequencing technologies for the neurovirulence test. And of course, we would like to welcome you all in a face-to-face final conference of all the activities that we are running in these years in Bangkok next December 2025. You will receive some information about this upcoming event. It's going to be a face-to-face, and we would like to see you all, if you can, travel to Bangkok and discuss with us about the future of the implementation of the MIT, the recombinant factor C, recombinant technologies, and other in vitro opportunities for biologicals. Again, thank you very much for being with us for this long workshop, but hopefully we were able to give you additional positive case studies and experiences about the feasibility of the implementation of the monocyte activation test. Thank you very much again to you all, and have a very nice rest of your day. evening or rest of your day. Thank you very much to you all. Goodbye.
Thank you, Laura. Ciao.
Thank you, Laura. Bye.
Goodbye.
Thank you.
Thank you.
Bye.
Thank you, Laura.
Thank you, all of you. Have a nice day. Bye.